Rukari sir, am I audible?
Yes.
Okay, so shall we start, sir?
Yes, ma'am. You can.
A very fresh good morning to all the dignitaries, guests, delegates with a great joy and immense exultation. I, Ms. Namita Bhosle, Assistant Professor, Department of Pharmaceutics, on behalf of IQAC and Savin Club of Yashwantrao Bhosle College of Pharmacy, extend a very warm welcome to all of you for the one-day national level virtual symposium on recent advancement and future outlook in novel drug delivery system. We are proud to announce that this national symposium is organized in collaboration with Association of Pharmaceutical Teachers of India, Maharashtra State Branch, that is APTI MSP. First of all, I would like to welcome our Executive Chairman, Honorable Mr. Achyut Savan Bhosle, sir. Heartiest welcome, sir. The President of Sri Yashwantrao Bhosle Education Society, Honorable Advocate Mrs. Asmita Savan Bhosle, ma'am. The Secretary of Sri Yashwantrao Bhosle Education Society, Honorable Mr. Sanjeev Desai, sir. I would like to welcome our Administrative Coordinator, Honorable Mrs. Sunitra Patak, ma'am. Heartiest welcome to all of you. We feel honored to have with us our Honorable Chief Guest and our today's keynote address speaker, Honorable Professor C.D. Upasni, sir, the President of APTI MSP. I would like to welcome our resource person for technical session 2, Honorable Dr. Bhushan Rani, sir, the Professor and HOD of Department of Pharmaceutics from Shri D.T. Vispute College of Pharmacy, Navi Mumbai. I would like to welcome our resource person for technical session 3, Honorable Dr. Satish Polshettiwas, sir, the Professor and HOD Department of Pharmaceutics from MIT WPU Pharmacy, Pune. Today, we are privileged to have an opportunity to listen from you all. So, on behalf of all gathered here, I welcome you all to this national symposium. I would like to welcome our beloved Principal of YBCP, Honorable Dr. Vijay Jagtap, sir, and the convener of today's national symposium. I would like to welcome respected Mr. D.M. Shinkar, sir, the HOD of PG Department, and the organizing secretary of today's national symposium. I would like to welcome respected Mr. T.G. Rukari, sir, the HOD of Department of Pharmaceutics. I would like to welcome the principal of Yashwantrao Bhosle College of D Pharmacy, Honorable Mr. Satyajit Sathe, sir. And finally, on behalf of the entire YBCP fraternity, I would like to welcome all the faculty members, participants from different sectors of India to this one-day National Symposium 2021. It is said that education is the most powerful weapon we can use to change the world. And therefore, the symposium has been arranged with the goal to strengthen the grit and determination of pharma persons to reach the excellence in the recent advancement. Ladies and gentlemen, we know that knowledge removes ignorance just like light removes darkness. And it is also said that if you lit a lamp for someone else, then it will brighten your path also. Even though we have arranged this national symposium on a digital platform in line with social distancing norms due to this COVID-19 pandemic, we can't forget our tradition. So let's start this event by virtual lamp lighting ceremony as a tribute to Mother Saraswati, the goddess of knowledge. And for that, may I request to Ms. Aishwarya Parab ma'am, Assistant Professor, Department of Pharmaceutics, to proceed with virtual lamp lighting.
कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तुते दीपज्योतिर्नमोऽस्तुते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः
Thank you so much, ma'am. Now, moving further, may I request to HOD of PG Department of Pharmaceutics, Honorable Dr. D.M. Shinkar, sir, and the organizing secretary of today's event to give a brief outline and introduction of today's National Symposium. So, please.
Yes. Very good morning, respected dignitaries and the resource person for today's symposium and all these delegates present here. Thank you for joining us at the One Day National Virtual Symposium 2020. It's my great privilege to walk you through the symposium. Today, we have eminent personalities as a resource person from industry and academics across the field who will be staying with us for whole the day, sharing their thoughts, knowledge, and opinions with more than 200 delegates from the different pharmacy institutions across the country. I welcome all the eminent speakers and chief guests from all over the country from different walks of life. We have come here to share your knowledge, thoughts, and vast experience with our participants. The theme of this symposium is on recent advancements and future outlook in novel drug delivery systems. We are proud to announce that this symposium is being conducted in collaboration with Association of Pharmaceutical Teachers of India, Maharashtra State Branch. The major outcomes of today's symposium are all the participants will get insights on recent developments in the field of NDDS and current challenges and possibilities in modeling a new drug development and breakthroughs in the drug development process. Also emphasize on future prospects and importance of NDDS in coming era in the pharmaceutical field. We are honored to have today our today's chief guest of this symposium, Professor Dr. C.D. Upasni, President APTI Maharashtra State Branch, who will be guide and motivate all of us in a few minutes in his keynote address. Throughout the day, we are in touch with eminent personalities on the topics in 4 sessions. Session 1, Dr. H.S. Mahajan will deliver a talk on nanotechnology, best novel drug delivery system. Session 2, Dr. Bhushan Rane will emphasize on recent advancements in novel drug delivery system. Post lunch, we have session 3 will be conducted by Dr. Satish Polshettiwar on topic bioinformatic tools and approaches in drug design and development. Last session will be delivered by Dr. Harshad Kothaude on role of regulatory affairs in formulation development. I would like to conclude my introductory speech by encouraging the delegates to participate with enthusiasm and discussions through the digital platform for the day in all sessions. I wish everyone a successful, safe, and fruitful symposium. Thank you.
Thank you so much, sir, for such a brief introduction. Now let's have the pleasure of listening to the opening and welcoming remark. And for that, may I request Honorable Dr. Vijay Jagtap, sir, the founder principal of Yashwantrao Bhosle College of Pharmacy, Sawantwadi since 2015, and convener of today's National Symposium, to deliver the welcome address. Sir, please.
Yeah, thank you very much, Ms. Namita ma'am. Very good morning to everybody. Sri Yashwantrao Bhosle Education Society's Honorable Executive Chairman, Mr. Achyut Sawant Bhosle sir, Honorable Chairman, Advocate Mrs. Asmita Sawant Bhosle ma'am, Honorable Secretary Mr. Sanjeev Desai sir, Honorable Administrative Coordinator Srimati Sunitra Pathak ma'am, today's guest of honor, Honorable Keynote Speaker and Chief Guest, Professor C.D. Upasni sir, President, Association of Pharmaceutical Teachers of India, APTI, Maharashtra State Branch. Honorable Technical Session Speaker, Dr. H.S. Mahajan sir, Department of Pharmaceutics, R.C. Piper, Shirpur. Honorable Technical Session Speaker, Dr. Satish Polshettiwar sir, Department of Pharmaceutics, MIT Pune. Honorable Technical Session Speakers, Dr. Bhushan Rane, Department of Pharmaceutics, College of Pharmacy, Navi Mumbai. And Honorable Technical Session Speaker, Dr. Harshad Kothaude, Director, Head, Regulatory Management and Trade Compliance, Merck Life Sciences Private Limited, Mumbai. All the other distinguished and honored guests, eminent scholars, delegates, organizing committee members, other teaching faculties and non-teaching staff members, media friends, dear students, ladies and gentlemen, very good morning. It is obvious to feel happy to deliver the welcome address, and I am also excited to feel blessed about this role, as this national symposium is built with the intent of active and interactive learning organized by Yashwantara Bhosle College of Pharmacy, Sawantwadi, in collaboration with the Association of Pharmaceutical Teachers India, APTI, Maharashtra State Branch. First and foremost, I would like to welcome our beloved honorable patrons of today's event, and due to whom we have got this opportunity to have third national symposium in Sindhudurga district, Sri Achyut Samant Bhosle sir. He has wholeheartedly given support and platform to intervene this scientific research. I also welcome Honorable Chairman, Advocate Mrs. Samant Bhosle ma'am, for always supporting and encouraging the events. Welcome to the Honorable Mr. Sanjeev Desai sir for his continuous and friendly support in growth of Yashwantara Bhosle College of Pharmacy. I welcome you, sir. My sincere regards to the Honorable Administrative Coordinator, Srimati Sunetra Phatak ma'am. I, on behalf of Yashwantara Bhosle College of Pharmacy, acknowledge your continuous relentless effort in development of all the events. Yashwantara Bhosle College of Pharmacy has borne the mantle of excellence, committed to ensure the students their own space to learn, grow, and broaden their horizon of knowledge by indulging into diverse spheres of learning. In our endeavor to raise the standards of discourse, we continue to remain aware in order to meet with the changing needs of our stakeholders. And for this, we have organized every year one of the symposium. Unfortunately, due to COVID, we are meeting here with virtually for the third time. I still remember in 2019, we have taken the first symposium that was based on pharmacognosy and herbal title, that is novel trends in drug design and natural product chemistry. And it was conducted on the 2nd Feb of 2019. The same date we have carried on the 2nd Feb of 2020, that was drug discovery research in India, that is current state and future prospects, that was based on the pharmaceutical chemistry. And this year we are very much fortunate that we have got permission to start our new course, that is Master of Pharmacy in Pharmaceutics. And for that, head of the department, Dr. D.M. Shinkar sir, has been appointed. And under the guidance of him, we are conducting this third national symposium virtually on the recent advancement and future outlook in novel drug delivery system. So for this symposium today, we have chief guest Professor C.D. Upasni sir. Sir is the president of Association of Pharmaceutical Teachers of India, APTI, Maharashtra State Branch. I wholeheartedly welcome you, sir. Objective behind this is already said by the Dr. D.M. Shankar sir, so I will not repeat that. But I extend my welcome wholeheartedly to all the four technical session speakers once again. Dr. Mahajan, Dr. Paul Chintiwar sir, Dr. Rane sir, and Dr. Kothovde sir. And would like to say thanks for accepting to conduct this technical session. I'm really pleased to announce that more than 200 registrations have been registered by the students of different era of graduate, postgraduate, PhD students and faculty members. I really welcome you all. We hope that this symposium will act as a medium for all of us present here to ponder upon the topic of discussions, challenges us to strive towards and inspire us at the same time. So I take the privilege to welcome once again to all the coordinators, faculty members, students and esteemed audience who have gathered here from the diverse places across the countries. I hope that all of you will have most productive day of interesting and stimulating discussions. I sincerely wish this symposium will be a great success, and not only as an opportunity to share knowledge and experiences in higher education, but also at the beginning of long and fruitful cooperation and friendship amongst fellow educators devoted to the most meaningful and worthwhile task, teaching and training our youth who will shape our future. So I again extend very warm welcome to everybody present here. Thank you. Thank you very much.
So much, sir. Your welcome address set a perfect platform for our today's national symposium. Ladies and gentlemen, I would like to add that Dr. Jagtap sir has completed his MPharm and PhD in pharmaceutical chemistry and having 18 years of teaching experience. He has published more than 40 national and international paper publications as well as presentations. He has one patent in synthetic chemistry with the subject novel fluorobenzothiazole-sulfonamide. Thank you so much, sir. Now, being an ideal example of inspiration and motivation for all of us, I considered it a great honor to welcome our Executive Chairman, Honorable Mr. Achut Savan Bhosle, sir, to share his views and best wishes for our third national symposium. Sir, please.
बरं इनॉग्रेशन झालं ना हां
Achut sir, please unmute yourself.
Sorry, just I didn't say. Okay. So thank you for inviting me, and congratulations and best luck for third national symposium. That is after COVID situation, you arrange such a wonderful sessions and inviting all of us. Thank you for it, and best wishes to all of you. Thank you.
Thank you so much, sir, for your words of wisdom and encouraging us all the time. Ladies and gentlemen, today we have distinguished Honorable Mr. C.D. Upasani, sir, the President of APTI Maharashtra State Branch, as a keynote address speaker. I feel extremely honored to introduce our today's chief guest, Honorable Mr. C.D. Upasani, sir. He is Professor of Pharmacology and Principal at SMGB Shriman Sureshwada Jain College of Pharmacy, Nemit Nagar, Nashik. He has completed his MPharm in Pharmacology and PhD at MAMS. He has 26 years teaching experience. He has guided more than 52 MPharm students and 16 PhD students. His area of research is oxidative stress, diabetes, neuropathy, pain, and inflammation. He has published 72 national, 36 international. He has 3 patents, and he has authored 16 books. He has attended number of national seminars, workshops, conferences as a resource person. I would like to add some affiliations with universities and professional bodies. He is the chairman of Board of Studies in Pharmacology, Faculty of Science and Technology at Savitribai Phule, Pune University, Pune. He is the member of Academic Council of Savitribai Phule, Pune University, Pune. He is member of Research and Recognition Committee at Dr. Babasaheb Ambedkar, Marathwada University, Aurangabad, and SP Pune University, Pune. He is member of Board of Studies in Pharmacology and Pharmacognosy at Kavitri Bahinabahi Chaudhary, North Maharashtra University, Jalgaon. He is member of Board of Studies in Pharmacy at Punyashlok Ahilya Devi Holkar, Solapur University, Solapur. He is ex-coordinator, Faculty of Pharmaceutical Science, Savitribai Phule, Pune University, Ganesh Kind, Pune. And he is president of Association of Pharmaceutical Teachers of India, Maharashtra State Branch, that is APTI. He is life member of Indian Pharmaceutical Association, Indian Pharmacological Association, life member and EC member of APTI, International Society of Heart Research, International Academy of Cardiovascular Science, and Heart Failure Society of India. He is life and EC member of Indian Pharmacy Graduate Association and the MSU Pharmacy Alumni Association. He has organized number of conference, workshops, seminars, and symposium at national and international level. I would like to say some awards and prizes of Sir. He is honored as member of National Academy of Medical Science by National Academy of Medical Science, New Delhi, during 53rd conference of NAMS at AIIMS Jodhpur in October 2013. He is honored with prestigious N.N. Datta Prize for Best Paper National Level Prize by Indian Pharmacological Society, conferred during IPSCON 2012, the annual conference of Indian Pharmacological Society held at Srimati Kishoritai Bhoyar College of Pharmacy, Kamki, Nagpur in January 2013. He is honored with the prestigious Bharati Vidyapith's Pune College of Pharmacists Pharmacy Teacher of the Year 2019, the National Level Award by Association of Pharmaceutical Teachers of India, conferred during 24th Annual National Convention of Association of Pharmaceutical Teachers of India, held at DIT University, Dehradun in October 2019. He has achieved Ms. Kanan Ishwar Mehta Memorial Oration Award 2020, conferred by Maharaja Sayajirao University Pharmacy Alumni Association, Vadodara, on 6 February 2021. He is conferred with the prestigious Best Principal Award, Professional College Rural, for academic year 2020-21 by Savitribai Phule Pune University on 10th February 2021. Ladies and gentlemen, today we have such an inspiring and dynamic personality with us. So I would like to request Honorable Professor Mr. C.D. Upasni sir to deliver a keynote address.
Good morning, all. Honorable Sri Achyut K. Sawan Bhosle, who is Executive Chairman, Advocate Asmita Ji Sawan Bhosle, Chairman, Sri Sanjeev Desai sir, Secretary, and one of my associate and my very good friend, Dr. Vijay Jagtap, who is the principal of Bhosle Knowledge Cities, Yeshwantrao Bhosle College of Pharmacy, Sawantwadi. And the person who made me aware about this activity and who invited me for this activity, Dr. Dattatreya Shinkar, who is my close associate from so many years when he was working in around Nasik district. So I'm thankful to all of you for calling me here as a chief guest and keynote speaker for recent advancement and future outlook in novel drug delivery system. I'm very happy to see the list of resource persons: Dr. Hitendra Mahajan, Dr. Satish Polshettiwar, Dr. Bhushan Rane, and Dr. Harshad Kothawde. All are closely associated with me. We are working together for this pharmacy education in Maharashtra region. I'm very much thankful to Namita Bhosle ma'am also for giving a elaborate introduction about me. What are the recent advancements? Because all of us are going through this COVID-19 pandemic from last 1 year. And this coronavirus family is troubling the humanity since a long time. And this is a newer version, detrimental version of coronavirus we are come across, and we are facing that problem. So I was thinking what to tell in this keynote address. So looking at your topic, recent advancements. So recently, all the scientists, all the pharmaceutical industries, all the academic institutions are working to get rid of this coronavirus, to reduce the detrimental effect of this coronavirus, and to improve the life and lifestyle of human being. So for this, many of the issues can be addressed. I know very well Dr. Satish Polshettiwar, who is a versatile dynamic speaker, and he will definitely address the issues related to this very well. Bhushan and Hitendra will talk about the formulation development aspect, and Harshad will talk about the regulatory aspect. But we being academic institution, we being teachers, we are not that fixed, or we are not that placing ourselves in the frame of time to develop the drug and develop the formulations. We being academics, we being in active academics, and we are having a source of highest energy. The youth, the young people, our students, these people are having a very high energy level. And if we being a teacher can motivate them, can guide them properly to the betterment of mankind, so definitely every academic institution will come up with some newer ideas, and by taking the help of existing ideas, we can definitely frame out better future outlooks in this novel drug delivery system. Why I am telling you all these things, there is a basis in this corona pandemic. Everybody was talking about this pandemic, the situation of human beings in this, the situation of society in this, and suddenly one particular segment came forward, and that segment, we know it very well as immunoboosters. Now, immunoboosters in the form of available medicine, marketed medicine, immunoboosters in the form of our Ajibai ka Batwa, that is our grandmother and our ancestors, whatever they are telling. So, Ayurvedic proprietary medicines and immunobooster in the form of food also. If you look at the sell or market position of these things, immunomodulator drugs, which are FDA approved category, immunomodulator Ayurvedic proprietary medicine, and immunomodulator food. Modulator means these are immunostimulants. Everybody has suddenly came into so much of consciousness of immunity, right? We have started yoga, we have started pranayam, we have started eating chawanprash, we have started eating Ayurvedic herbs, everything. So I would like to tell you some insight or some idea about what this is. You know, respiratory system, it is a vital system and it is very delicate system also. Respiratory system gets frequent attacks by microbes every now and then, allergens every now and then. I think every one of us must be suffering from common cold, coughing, sneezing at least once a year. Look at the population of India and look at the people, those who are suffering from these respiratory problems. So I feel that our academic institutions, our teachers should focus on this, because if you want to test the immunomodulatory or immunostimulatory type of effect, there are no much equipments are required for that. There is no very high cost investment of very high cost material. These are very simpler methods, very tiny methods which are available in the literature. And if we can guide ourselves, our teachers and our students to work on this immunoboosting type of testing of immunoboosting material, immunoboosting formulation, immunoboosting Ayurvedic formulations, which is there in our literature from hundreds of years. I tell you, this global market of food products is nearly about $8,30,302 million in 2019. And it is having growth around CAGR 3.8% annually. And it is consistent since 2015. And it is expected to grow at the rate of 6.2%. So it will go nearly 10,56,384.7 million by 2023. We are in 2021. We, the teachers, we, the students of science and pharmacy, we must come together, work on a particular idea. Dr. Vijay Daktab, I would like to tell you, you are located in Konkan region. Konkan region is rich in medicinal plants, material. So, if we can gather some of the material and if we can formulate something out of that, your institute can have hundreds of patents of combining those material and forming a particular formula if it is tested scientifically. So a very simple immunity testing methods are there. Simple, very normal instrument you can purchase in your institution, and you can form a team of 1 teacher and 10 students together. They will be working on one particular formula and they will be testing. There is a cell testing available, there is a cell culture testing available, and very few in vivo type of methods are there where you can have a blood sample and you can go for the measurement of neutrophil, eosinophil and other things as far as immunity is concerned. So in this view, these immunity boosters, the growth resulted in the material and the social awareness about these drugs, these formulations has increased to such a level that every industry, every academic institution which is working on this will be getting high returns in coming future. The recent medical dialogue, what has been taking place on with reference to this particular platform, that even a simple food habit, why Indians didn't suffer from this pathetic and dangerous COVID-19 situation. WHO has given at that time, the WHO has given the figure of 25 to 50 crores of Indians will suffer from COVID-19 pandemic. But what is the current situation? Till date, hardly 25 lakh people are lost their lives. In the population of 135 crore, only 25 people are getting affected. What is the percentage? Right? You look about the percentage. The fact that the role of our Indian food habit has played a vital role in protecting the human beings against this COVID-19 pandemic situation. I tell you, every area of India is having a typical type of food habit. If you come in north side of the Maharashtra, food habit is different. In Konkan, the food habit is different. In Vidarbha, there is a different food habit. In Madhya Maharashtra or in Paschim Maharashtra, there is a different food habit. But one thing is very common in this. We are taking green vegetables in our food. It is compulsory, like every day we go for a fruit vegetables or green vegetables. We eat green a lot. What is the theory they have placed to support the statement that why India didn't suffer a lot from this COVID-19? The theory is something like this. They have correlated our food habit with gut microflora. Our immune regulation and homeostasis. Metabolic functions of prominent bacteria form their ability to produce essential vitamins, then metabolites and fermentation processes. which are there in our intestine. For example, fermentation of dietary fiber, whatever fiber we eat from this green vegetable, fermentation of this dietary fiber by gut microbe result in the formation of short chain fatty acids that are known to regulate the immune response via G protein coupled receptors, that is G protein coupled receptor 43 and 120. How interesting this is. Food, whatever we are eating, it may be a fenugreek leaf, or it may be a palak leaf, or it may be anything, any green vegetable, whatever we are eating every day. That green vegetable is getting processed in such a way that these intestinal microorganisms, they are converting it to the short chain fatty acids. And this short chain fatty acid is responsible for regulating the immune response. And therefore, worldwide it is accepted, and it is nowadays it is talked very loudly that Indians, they have a better immune system and better immunity as compared to Western world. So this COVID-19 pandemic has started these dialogues, and the concept, this particular concept provides a rationale for use of probiotics as a modulator of immune system. I will request all the delegates and all the teachers, those who are listening to this, attending this seminar. We are talking on national level that use of these materials in a rational proportion. Every institute, if works on this, every institute will have a patent, and every institute can share this formula with the pharmaceutical industry, Ayurvedic industry, and they can come up with a nice product. And this product will be helping the mankind, helping the human society for boosting their immunity, and that will help everyone to fight against this coronavirus pandemic. So this is the, I will not go into the detail of that. I can talk about hours together on this, but it has given a click to me that if we pharmacy teachers, if head of the institution like Dr. Jagtap or myself, if we can motivate our faculty, motivate our students to form a quality circle scientific groups, and if they work on one particular formula, that will definitely help the society also, the institute also. It is a win-win situation for all. If you go through the syllabus, final year syllabus, there is a project given in that syllabus. Right? Every final year student has to undergo some kind of a project activity. In this project activity, if we can guide some of the students to work on this formula, some of the students to work on the market survey, some of the students to work on the formulation development aspect of this, this will definitely bring a new enthusiasm, new energy level will be involved in this, and every institution will come up with a novel idea, and that will be a real future outlook as far as the market or a cell or a pharmaceutical industry as far as probiotics are concerned, as far as immunoboosters are concerned. And this is the thing which is not being tested scientifically from so many years. Very few people are working on very few type of formulation. Even I tell you the example of that arsenic 30 album tablets what are been distributed to lakhs of people. But do we have a scientific report of that? Whether yes, it is really doing that miraculous act. So why can't it, the idea came in our mind, why we should not work on that to give a scientific basis to the existing knowledge. Whole world is talking about repurposing of drug. So why not to talk about repurposing of formulations also? Drug formulation is a media by which drug is given to the patient or the people. So why not to talk about repurposing formulation? In this region, Nasik and Malegaon region, the Unani Institute has formulated one particular mixture of a powder, and that mixture of a powder is known as Malegaon ka Kala. That powder is given to everyone. That powder has to be boiled, and then you have to drink that decoction twice a day. But it is claimed that this Malegaon ka Kala has treated and has cured so many people that the corona count in Malegaon city has drastically reduced because of this Kala. But what this Kala is? This Kala is a mixture of 5-10 herbal products. These are being crushed, powdered and mixed together, and it is given for decoction purpose. So this is also one of the idea that if this decoction is being tested for stability and if formulated very well, and if it is ready to drink, decoction is available to the market, it will be a miraculous thing. And many pharmaceutical industries, especially those who are working with Ayurveda and Ayurvedic formulation, they will be coming up with this idea. And it will be our duty, all the head of the institutions, all the teachers and all the students to look for such opportunity and try to get involved in this active type of pharmaceutical research. You never know, like a product like Liv 52 can have a US FDA approval, and that is the only medicine till date considered after Silymarin for the liver disorders. So like this, some of the institution or some of the groups, those who are working on this, may come up with a scientific formulation of this material for COVID-19 and likewise. Nowadays, respiratory system is a very important system as far as research is concerned. Everybody is working on that. Whatever Covishield and other vaccines are made available for the society, some of the scientists are working on the nasal delivery of that vaccine. I remember while doing my PhD, one of my friend, he worked on oral delivery of the tetanus toxoid and diphtheria vaccine, and he could succeed in that. The research made for oral delivery of tetanus toxoid vaccine has been taken up by WHO, and WHO is giving guideline based on that research. This was a big achievement of pharmacy department, MS University, Baroda. So likewise, everybody is working on his own idea. So we are the teachers, we are the students, and we have a very free time available, liberal free time available to think on these logical approaches and come up with idea. I'm sure today, Dr. Hitendra Mahajan, Dr. Satish Polshettiwar, Dr. Bhushan Rane, and Dr. Harshad Kothawade will guide us, guide our students, guide our delegates, and guide us also to how to achieve this future outlook goals as far as the pharmaceutical development is concerned. I'm thankful for the organizers. I'm thankful to Dr. Jagtap and Dr. Shinkar for calling me here. Thank you. Thank you very much. God bless you.
Thank you so much, sir, for your influential message and sharing your great knowledge with us. We will definitely follow your suggestions in future to deal with such pandemic situation. Thank you so much. Now may I request to our principal, sir, Honorable Dr. Jagtap, sir, to give a brief introduction about your institute by sharing a video.
Yeah. You're getting voice? Audio? Yeah. Just a second. Actually, I have shared it, okay? Can you see my desktop now? Okay, let me check. Can you see now? Yeah? Oh, 1 second. What is right now there? Okay, that's good. Okay. Now? Yeah. So audio? I think that is disabled by you only. Okay. Okay, let it be video.
Sir, optimize the video. Sir, optimize the audio. The option is available. Click on the right hand side. Optimize the your audio video.
Let me check.
On Zoom, actually. On Zoom, there's a, yeah. Click on that setting. Okay. It is available.
Tushar, I think that, yeah, you can do that, okay? You enable the audio.
Tushar, sir. Sir, while sharing screen, there are two small options on the right side.
Go to the settings.
Sir.
Okay, you can see the video now.
Yes, sir. You can stop your sharing and again, once you
okay, I stop sharing
and then go for the sharing again. I mean,
okay.
At the bottom there are two options. There are two square. You have to mark them.
Okay, yeah, that's good. Okay. Yeah, thanks. Let me know.
उच्च शिक्षणासाठी
Yes, sir.
मुंबई पुणे अशा मोठ्या शहरांकडे धाव घ्यावी लागत होती. याच अस्वस्थतेतून उदयाला आलेल्या विचारांमुळे इथल्या गरजू विद्यार्थ्यांसाठी विद्यादानासारखं महत्त्वपूर्ण आणि पवित्र कार्य हाती घ्यावं असं अच्युत सावंत भोसले यांनी ठरवलं आणि विद्यादानाच्या क्षेत्रामध्ये पदार्पण केलं. अल्पावधीमध्येच बी फार्म आणि डी फार्म अभ्यासक्रम इथे सुरू करण्यात आले. यापैकीच बी फार्मसी कॉलेज हे सिंधुदुर्ग जिल्ह्यातील पहिलंच डिग्री फार्मसी कॉलेज आहे आणि हे कॉलेज मुंबई विद्यापीठाशी संलग्न जिल्ह्यातलं एकमेव कॉलेज आहे. काळानुरूप प्रगतीपथावर वाटचाल करताना संस्थेमध्ये अनेक बदल घडवत कोकण प्रांतातील विद्यार्थ्यांना उच्च शिक्षित करणं हे आमचं ध्येय कायम आहे.
फार्मसी कॉलेजविषयी सांगताना मला खूप अभिमान वाटतोय की हे सिंधुदुर्गातलं पहिलं मुंबई विद्यापीठाशी संलग्न असलेलं कॉलेज आहे. अदरवाईज आपल्याला मुंबई किंवा पुण्याला आम्हाला डिग्री कॉलेजसाठी जावं लागत होतं. हे एस्टॅब्लिश केल्यानंतर लगेच आम्ही दुसऱ्या वर्षी फार्मसी डिप्लोमा कॉलेज चालू केलं आहे. 2016ला पुन्हा डी फार्मसी कॉलेज चालू केलं जी इथल्या पुन्हा ह्या कॅम्पस सावंतवाडीसाठी गरज होती.
We value innovation, learning and the advancement of knowledge. We value research and its application toward improving the use of medications in society. We value pharmaceutical care and professionalism. We value diversity in all its form and we value our students, faculty, staff and friends like you.
लॅबोरेटरीज आम्हाला चांगल्या प्रकारचं प्रॅक्टिकल ज्ञान देतात आणि हे कॉलेज फक्त इथपर्यंतच न थांबता आम्हाला अजून कल्चरल ॲक्टिव्हिटीज किंवा सायंटिफिक ॲक्टिव्हिटीज जर असतील तर तिथे पण ते आम्हाला पुढे करते. सिम्पोजियम म्हणा किंवा पोस्टर प्रेझेंटेशन असेल. नेहमी कॉलेजची वर्षातून एकदा हॉस्पिटल व्हिजिट असते, परत इंडस्ट्री व्हिजिट असते. तर हे सगळे आमच्या प्रोफेशनल स्किल्स असतील, कम्युनिकेशन स्किल्स असतील, ते सगळे आम्हाला इथे वाढवायला मदत करतात.
We have our vast library which includes more than 1,500 reference books along with the online and printed journals as well.
विद्यार्थ्यांचं संभाषण कौशल्य वाढावं, त्यांचा व्यक्तिमत्व विकास व्हावा यासाठी निरनिराळी व्याख्यानं आणि प्रशिक्षण कार्यक्रम आयोजित केले जातात. विद्यार्थ्यांसाठी वसतिगृह, मेस, कॅन्टीन, भव्य क्रीडांगण, व्यायामशाळा, बस सुविधा, सीसीटीव्ही यंत्रणा अशा सुविधा भोसले नॉलेज सिटीच्या परिपूर्णतेमध्ये भर घालतात. त्याचबरोबर विद्यार्थ्यांच्या आरोग्याच्या आणि सुरक्षेच्या सर्व उपाययोजना इथे करण्यात आल्या आहेत. निसर्गानं भरभरून सौंदर्याचं दान दिलेल्या कोकण प्रांतामधील भोसले नॉलेज सिटीचा हा कॅम्पस व्यावसायिक आणि तंत्र शिक्षणाद्वारे विद्यार्थ्यांसाठी व्यवसायाच्या जास्तीत जास्त संधी उपलब्ध करून देणं, भविष्यात उपयुक्त ठरेल अशा प्रकारच्या शिक्षणाचं संशोधन करणं, विद्यार्थ्यांना सामाजिक जबाबदारी, मानवी नीतिमूल्य आणि स्वतःच्या अस्तित्वासंदर्भात सजग करणं ही या संस्थेची ध्येय धोरण आहे. शिक्षण, संशोधन आणि विस्तार या 3 मूलभूत तत्वांवर आधारलेली ही संस्था प्रगतीच्या उन्नत मार्गावर कार्यरत आहे.
Thank you so much, sir, for sharing such a beautiful video of Bhosle Knowledge City. Now I would like to request respected Mr. Tushar Rukari, sir, HOD Department of Pharmaceutics and coordinator of today's National Symposium to conclude this inaugural session.
सर बोला. हो हो.
Thank you so much, sir. Ladies and gentlemen, this is the time to welcome and introduce our today's resource person. First of all, I would like to thank and welcome all the resource persons who have come to share their knowledge and experience with all of us to ensure an outcome that is grounded in the concrete realities of this national mechanism. Sir, shall we start with the technical sessions? Rukari sir, shall we start with the technical session?
रिपीट आलेला म्हणजे ग्राममध्ये सेम मग आता हे अंडर साईज कसे घेऊ पर्सेंटेज
Just a minute, we will take the screenshot as a proof of this event. I would request all the participants to turn on your videos. I request all the participants, please mute yourself.
Namita ma'am, unshare that PPT, okay?
Okay, thank you so much all. Now we are entering into our first technical session of today, which is based on nanotechnology based novel drug delivery system. The resource person for this technical session is Honorable Dr. H.S. Mahajan, sir. To chair the session, may I request to Ms. Shweta Shirodkar, ma'am, Assistant Professor, Department of Pharmaceutics, to take the opportunity to introduce our today's resource person, Honorable Dr. H.S. Mahajan, sir. Over to you, ma'am.
Am I audible, ma'am?
Yes, ma'am, you're audible.
Thank you very much, Namita Bhosle, ma'am. First of all, very good morning to everyone. We all are here for one-day National Virtual Symposium on recent advancement and future outlook in novel drug delivery system. It's my pleasure to welcome you all for the first session of this symposium. I'm very much thankful to Principal Sir and event coordinators for giving me this opportunity, and it's my privilege to introduce resource person of first session, Honorable Professor Dr. Hitendra Mahajan, sir. Presently, he is working as a professor and head of Department of Pharmaceutics, R.C. Patel Institute of Pharmaceutical Education and Research, Shirpur. He has completed PhD at North Maharashtra University, Jalgaon, on the topic formulation and evaluation of nasal mucoadhesive drug delivery system. He is having 18 years experience in teaching and research. He is recipient of many awards and honors, like he is recipient of Professor Ambika Nandan Misra Research Excellence Award in Pharmaceutics and Drug Delivery Award in 2019 by Association of Pharmacy Teachers India. Then he is recipient of Research Initiative Award 2016 by Sunpure Research Incubation Center. Also, he has received Research Award by UGC in 2014 and 2016 and Srishti Appreciation Award by Biotechnology Industry Research Assistant Council in 2017. Also, he is recipient of Indian Drug Best Research Award in 2017 by Indian Drug Manufacturers Association. He has also received award of Indian Pharmaceutical Association Best Research Presentation in 5th and 6th Symposium on Nasal and Pulmonary Drug Delivery in the year 2011 and 2013. He has also received first prize at university in Avishkar in the year 2006, 2016 and 2017. He is principal investigator of projects which are sponsored by AICTE, UGC, Biotechnology Industry Research Assistant Council, Science and Engineering Research Board, Department of Science and Technology, RGST and Industry, worth rupees 121.99 lakhs, based on different topics like nasal mucoadhesive drug delivery system, nanoemulsion, polymeric micelles, vitamins, multivitamin formulations, novel anti-malarial formulations, minimal invasive microneedles for diagnosis and treatment of ocular tuberculosis, etc. He has worked as a reviewer to access a research proposal submitted to the Ministry of Science, Technology and Space of ISRIL, A&D Department of Science, Technology of India. He has acted as expert for evaluation of Avishkar 2017 and 2018 in North Maharashtra University, Jalgaon. He has also published 29 research articles in international journals and 61 research articles in peer-review journals. He has also participated and presented 14 research papers in various national and international conferences. He has also published 3 books of the subject biopharmaceutics and pharmacokinetics, pharmaceutical management, and physical pharmaceutics too. He is having 2 Indian patents on his name. He is also ad hoc reviewer of various international journals like Drug Delivery, Pharmaceutical Development and Technology, Journal of Microencapsulation, Liposomal Research, Carbohydrate Polymers, Material Science and Engineering, International Journal of Biological Macromolecules, etc. He has also attended and worked as coordinator of national conferences, workshops, and faculty development programs, and of AICTE-sponsored seminars. He has guided 3 PhD students, and currently 8 PhD students are registered under his guidance. He has supervised 72 MPharm projects since 2007. He has also delivered 16 guest lectures, webinars, and expert talks at different colleges around India on the different topics like different funding agencies and writing research projects for funding, mucoadhesive drug delivery systems, micelles as nano-sized carriers for cancer treatment, nanomaterial in drug delivery, controlled and site-specific ocular drug delivery, nanoparticles, validation aspects of sterile process, nano-emulsion, bioavailability enhancers of herbal origin, preparation and documentation of National Board of Accreditation. He has also worked as evaluator, chairperson, and expert for scientific sessions at national conferences, SPTM Shirpur, Avishkar, and also in national symposium at different colleges. Also, I would like to add in his introduction that he is also life member of Indian Pharmaceutical Association and Association of Pharmaceutical Teachers of India, member of All India Council for Technical Skill Development, and also Pharmacy Council of India Inspector Panel members. I would not take so much time. Just a note to all the participants, after the session, we will have question answer session. So you can put your questions in the chat box that can be addressed by Professor Dr. Mahajan sir at the end of the session. Professor Dr. Mahajan sir will deliver the session on the topic nanotechnology based novel drug delivery system. With this brief introduction, now I would like to request Professor Dr. Hitendra Mahajan sir kindly proceed for the session. Over to you, sir. So please.
Okay. May I audible?
Yes, sir.
Okay, thank you. Okay, good morning one and all. I'm thankful to Ms. Shweta ma'am to giving my detailed introduction to all delegates. It's too detailed. Actually, she said I'm giving a short introduction about this one, but it is very lengthy. On outside, I'm most thankful to Dr. Vijay Japta and Dr. Shinkar for giving me an opportunity to share my some research work based upon nanotechnology in a novel drug delivery system application is that. I'm most thankful to Dr. C.D. Upasni for his motivational keywords. And during these keywords, he said that we must think about the repurposing of the drug and repurposing of the formulation is that. Now, today, what we are going to discuss right now, that is nothing but the repurposing of drug delivery system is that in a nano formulation forms. Now, once we are talking about the nanotechnology based novel drug delivery system, already the concept of nanotechnology is known to all of us, and the concept of novel drug delivery system is also known to all of us. Once there is some lacuna is to be there in a conventional drug delivery system, and to overcome this one, we are shifted to the novel drug delivery system, and still there is some limitations of the novel drug delivery system is there. That's why there is some applications of nanotechnology has been incorporated into the nanotechnology, into the novel drug delivery system. And by this way, the attempts has been made to improve the performance of the novel drug delivery system. Now, once we're talking about this nanotechnology based drug delivery system, novel drug delivery system is that, then everybody should have some idea about the nanotechnology. Now, in today's session, whatever our agenda is there, we'll have some background of the nanotechnology in terms of history. Then we'll try to know about the what exactly the nanotechnology is there. Then what are the different nano carriers currently being used in a drug delivery system? The background of the different types of nanoparticulate based systems which are commonly to be used. Then there are certain limitations once you are going for any newer techniques are there. Every technique is having these pros and cons are there. Therefore, there is one limitation of this nanotechnology based system, that is nanotoxicity, which is major concern is that, and the measurement of the nano size. This is very critical aspect is that. Once we are talking about proving the case that but the system which is to be developed really it is a nano or not is that, then we'll see the application aspect of the nanotechnology based novel drug delivery system, and I will share my one research work in a briefly very where we made application of this nanotechnology for improving the performance of the novel drug delivery system. Now, moving to the history, the nano concept of nanotechnology that has been very well documented in ancient literatures are there, and since 9th century there is some documentary evidence out there about the presence of the nanotechnology in an area of Mesopotamia where there is some pots, ancient pots are there that are coated with the gold particles are there, and they are due to the presence of this small size in a nanometric range, these gold particles are producing some kind of the luster, and that's why it appears like shining material is there. And once there is a in a museum once they are placed for some longer duration is there, the shining is remains same. That is the history about the nanotechnologies to be there. But at that time might be the absence of the some characterization techniques for the nanoparticles. But after some time in a current era, due to the availability of the particle size determination, it has been proved that what are the gold particles which are nanoscale, having the range between the below the 100 nanometer is there, they're coated on these spots are there, and that's why it is showing the luster is there. If you are talking about the use of the nanotechnology in a drug delivery system is there, then in a Indian literature, Indian system of medicine, the basmas are there. Basmas are nothing but the Indian nanomedicines are there. And once we are talking about the Indian systems are there, either because of the lack of knowledge, we are not aware about the things are there. But once nowadays we are having some technology with us to characterize the number of nanoparticulate based systems are there, one can clearly say that what are the basmas previously being used or previously been reported by our ancestors are there for the treatment of some diseases, that basmas are nothing but the nanomedicines which are previously been used by the Indian scientists are there. And we can say the Indians are the pioneer in a nanotechnology in a drug delivery system is that. Then when the first time the idea about the nanotechnology has been proposed or it came into the scientific domain is that this has been proposed by the Richard Feynman, who is the physicist by profession, and he is the recipient of the Nobel Prize for his work on some quantum mechanics is that. And during his one session in 1951, first time he talked about the nanotechnology concept is that, and he written as one book. There is plenty of room at the bottom is that. In this book, he is right about that there is a somewhat is present into the flask at the bottom that is not to be characterized, but that he terms the case it is a nanomaterial which are not to be seen by the naked eyes are there. That possibility has been given by the Richard Feynman. That's why he is to be called as the father of nanotechnology in terms of physics is there. And what is the idea he is given? He considered that the possibility of direct manipulation of individual atoms as more powerful form of the synthetic chemistry than those used at that time is there. Because at that time there is a certain limitations are there with the scientists to characterize the materials are there, and due to some advances in microscopic developments are there or microscopical examination developments are after some time, after near about 20 years period or after 1951, once he proposed the concept of something is to be present which is not measurable one in terms of nanoparticles. He proved that concept in the form of the quantum dots are there, and the idea of nanotechnology was born after the some clues which are given by the Richard Feynman is there. Then what is the current status of this nanomedicine is there? If you see the distribution graph, out of 100% contribution of the nanotechnology industry focus is there. Only 8% focus is there on a medical or healthcare is there. A majority focus is there on hybrid materials for some daily use applications are there. 20% contribution of industry in terms of nanotechnology that is coming from the information technology. 17% coming from the semiconductor. Then 11% is coming from the nano MEMS, and other 10% are there. And only 8% of the contribution is coming from the medical and healthcare societies there. And what is the majority of the progress in nanomedicine is there? It's been in the use of nanoparticles that is related to the drug delivery is there. And very little contribution is again coming in terms of the nanotechnology based devices for some diagnosis of the disease is there. That type of the little contribution is there, but due to the advances in the technologies nowadays available with the industries are there. They are going for the increasing this contribution of the nanotechnology into the medical and healthcare system. Then what is the nanotechnology exactly? The first time the definition of the nanotechnology come into force according to the UK Royal Society and Royal Academy of Engineering. Nanotechnology is defined as the design, characterization, production and application of structures, some devices and systems by controlling the shape and size of that material to the nanometer scale, that is having one or more dimension of the order of 100 nanometer or less is there. Then once we are talking about the nanometric range is there, or less than 100 or less than 100 nanometer is there. Then what is exactly the 100 nanometer? It is the 1 billionth of the meter is there. Means 100 nanometer is equal, 1 nanometer is equal to the 10 raised to minus 9 of the meter is there. That much small particles are there. What is the definition which is given by the UK Royal Society and Royal Academy of Engineering is that is by considering the all applications. They are not taken into account only the healthcare or medicine or drug delivery application is there. Once we are talking about this definition is that in terms of dosage forms or for drug delivery system, this seems to be the insufficient definition in that. Say for example, if you are talking about the shape and size of to the nanometer scale having one or more dimension, as far as the drug delivery system has to be concerned, you should think about the dimensions of the product is that in order to complete absorption of the drug, complete uptake of the drug by the cellular membrane is there. One should talk about the shape of the material is there, and once we are talking about the 100 nanometric size is there, this is also not advisable. Whenever there is any exogenous substance entering into the body, which is having a very less size, then there is a phagocytosis will be occur, and that is to be taken as a exogenous substance, and there is a provoking of the immunological response will be occur is that. That's why this definition needs to be modified in order to meet the requirements of the drug delivery system is that. Then what could be the appropriate size for the drug delivery system? You can go up to the 500 nanometers are there. Then it will not be term and it should be round in a shape ideally for the drug delivery application is that. Then in order to avoid such type of the immunological response, which is to be commonly to be occurred towards the some exogenous substances are there, there needs to use such type of the carriers to prepare the nanoparticles are there or nanotechnology based drug delivery system that should be mimic to the endogenous substances of the body, which are hydrophilic in nature, which are having some structural similarities with the what are the endogenous substances which are present in a body. Then and then only one can say that the nanotechnology based product can be easily accepted by the individuals as a drug delivery system. Now there is some different scales are to be there, and we are able to identify what exactly the nanometric size is to be there. If you see the meso scale size are there, say for example, if it is a 1 meter height is for the human being, 10 centimeters for the palm, and 1 centimeter, 1 millimeter. All these are the some meso scales are there. Up to the 100 micrometer, the size goes to the hair structure is that. And if you see the 100 nanometer size, which is less than the viruses is there, and it is in between the size of the hemoglobin and virus is there. That will be the appropriate size for the nanoparticles are there. But being these are the endogenous substances, hemoglobin 1, the body is very well equipped with the some immunological responses if it is externally to be administered, and once virus is to be enter, which is having the 100 nanometer size is there, there's an immunological response will be developed by the body. To avoid this type of the issues with the externally administered nanoparticles are there, one should try to increase the size in between this micro scale or above the 100 nanometer, or try to produce in the form of the some surface modified nanoparticles to avoid such type of the reaction of the body towards the nanoparticles or nanomaterials. Then This nanotechnology is nothing but the manipulation of matter at atomic or molecular scale to create the material with the new or advanced properties on it. By doing this nanotechnological manipulations, there are some changes has to be made into the atom or molecule in order to create the material with some improved properties. The improved properties is nothing but the either there's a change in adhesion of the drug to the or that nanomaterial to the cell membrane is there, and in this way there is a change in the uptake of the these materials by the cell membrane is there, and that's why there is a sufficient loading of the drug which is loaded in a nanoparticles into the cells will be possible in that type of the modifications are to be or manipulations are possible by using the nanotechnology, and that's why we are able to prepare such a drug delivery system that meets the individual requirements, individual patient requirement is there. And by doing this one, we are able to create individualization of the dosage form for the requirements of the individual patients. This nanotechnology integrate the medicine and engineering in order to help the quickly advance the world of medicine is that, and this is only the purpose of nanotechnology implementation in a drug delivery system, that whatever the conventional drug delivery system or advanced drug delivery system or novel drug delivery system what we call, they are having some limitations are there, and to have some specificity in a targeting to achieve the active type of targeting, the nanotechnology is doing well. By using some applications of engineering, we are able to have some improved performance of the such drug delivery system, and that will create some advances in the world of the medicine as well. Then following on some advantages of nanotechnology, as I already said, if it is a nano materials they are liable for some kind of the immunological destruction, phagocytosis, or some degradation in that. Then by using some kind of the specialized type of the carriers, we can protect these drugs from being degraded into the body before they reach to the target site. And in this way, there is a possibility to attain the specific targeting to the specialized target towards we are expecting the release of the drug in that. Then enhancement of the absorption of drug into the tumors and into the cancer cell themselves. Whatever the research work currently is to be going on in terms of the nanotechnology based drug delivery system is that is related to the improving the performance of the chemotherapeutic agents which are used in a treatment of cancer management in that. Therefore, once the nanotechnology based formulation containing anti-cancer drugs are to be prepared, then there is a chances of enhancement of absorption of drug into the tumor is there by doing some active and passive targeting is there, and that's why there is a improvement in a performance of the drugs is there, and avoidance of some limitations related with the chemotherapeutic agents are. Then it allows the better control over the timing and distribution of drug to the tissue, making it easier for oncologists to assess how will they work is that. That can be possible by doing the passive as well as the active targeting is that. And in this way, we avoid the unwanted exposure of the normal cells, which doesn't need any chemotherapeutic agents is that. And by this way, the performance or the toxicity of the anti-cancer agents can be minimized in that. Then to prevent the drug from interacting with the normal cells and thus avoiding the side effect is that is possible with the use of the nanotechnology based drug products on it. There are a number of limitations or we can say disadvantages of this nanotechnology in a drug delivery system. Since these particles are very small, the problem can actually arise from inhalation of these minute particles are there, because we can't prevent the presence of this particle in a environment is there, because they are not seen with the naked eyes are there. Therefore, there's a lot of issues are there regarding the unwanted inhalation of these minute particles, and they are precipitating some kind of the toxicities are there. In current scenario, if you are talking, the nanotechnology is very expensive thing is that, and that's why though there is a lot of nanotechnology based products are available, that are not available to the Indian population because of the cost issue is there, and the cost is nothing but the cost of the manufacturing is there. That is adding the cost of the product to the cost of the product, and that is creating the product is very expensive one. And another thing is that there is some certain limitations of the scale of operations in there. It is quite difficult to synthesize the nanoparticle in a successive batches in there and a larger scale in there, and that's why it is becoming somewhat more expensive in nature. As we are unable to transform the product into the commercially viable product is that they are creating some burden on lung cancer, mesenchymal tissue lymphoma, central nervous system, and genitourinary cancer treatment is that. All these are the developments which are going on, and what are the drugs, investigational drug under the clinical trials are there that are belonging to the anti-cancer agents are there. If you are talking about the Indian scenario, we are lacking very much in our nanotechnology based drug development. And if you see the Indian industry also not doing any that much extensive work related to the nanotechnology based products into the market is there. And since the materials which are given in the form of nanotechnology based dosage forms are there, that are ideally to be present in the form of the particles are there. And the particles are classified according to the diameter as nanoparticles which are having the 1 to 100 nanometer size. The fine particles are having the size between 100 to 2500 nanometer, and coarse particles more than 2500 to nanometer and above or below the 10,000 nanometer. These are the different size represented by the nanoparticles. Now, once we are talking about the nanoparticles for the drug delivery applications in there, then it should be the particle that is having the size ranging from 1 nanometer to the 500 nanometers. As we are already talking about the definition of the nanotechnology in a drug delivery system, if it is having a size more than 100 nanometer, then it is also acceptable for the drug delivery applications. Then what exactly the nanoparticles for drug delivery application and why it is to be prepared? Any particle that size between 1 and 100 nanometer in terms of diameter, that are to be called as the nanoparticles are there, and by different means of administration they are to be administered into the body. The purpose of preparing the nanoparticles of the drug substances, that is allow one to change the pharmacokinetic properties of the drug without changing the active compounds. Whenever the drugs are given in the form of nanoparticles, all the kinetic related process, absorption, distribution, metabolism, and excretion, the rate and extent of all these ADME processes get changed, and that's why there is a change in a concentration of the drug available at the site of action. That is going to be changed. Now, once we are talking about the any API which is to be used in a treatment of cancer, after its administration by any route of administration, it is undergoing some extent of absorption. Prior to reaching into the systemic circulation, it is undergoing some kind of the biotransformation that is called as the pre-systemic metabolism of the drug. Then it reaches to the systemic circulation, then it is distributed into the different organs are there, and then after distribution and metabolism, it is converted in the form of some excretable form. It is undergoing some elimination excretion process. By doing nanoparticulate based system or converting it into the nano materials are there is a changes is to be occur. The rate of extent of absorption get changed, the rate of extent of metabolism get changed, there is avoidance of first pass metabolism will be occur, there's the rate of distribution among the different body compartments get changed, the rate of elimination will be changes there. Sometimes the one can able to prepare the long circulating nanoparticles are there that are remain into the circulation for long period of time, and in this way we are able to extend the retention of the formulation or drug into the system until you are getting the desired pharmacological response. That's why the conversion of the drug in a nanoparticulate based system are definitely going to increase the pharmacokinetic properties, and that is the one of the purpose of the creation of the nanoparticles of the drug substances are. Then when we are talking about the definition of the nanotechnology, there are two aspects are there: size and shape of the material is there. All materials has a size, and all size matters are there. Like that, there is a nanoparticles are there, fine particles are there, coarse particles are there, and due to this, there is a lot of properties of the materials get changed. And the general properties of nanoscale particles are high surface area to the volume ratio, and due to the nanoparticulate size or nanoscale particles present, there is a high surface area to the volume ratio is to be provided, and due to this, the nanoparticles are able to interact with the biomolecules on the surfaces of the cell. They absorb the drug well through the lipoidal membrane subjected at the different absorption site, and able to diffuse through the body walls are there, and that's why they are easily permeable in nature. Is that one has been concerned? The spherical particles are easily taken by the cell, then the non-spherical ones are there, and therefore most of the particles which are to be used in a drug delivery system that are present in the form of the spherical particles are there, or the attempts has been made by the formulation scientists to keep it in the form of the spherical particles are there. Then various nano carriers currently being used, that includes the nanoemulsion, nanoshell, nanospheres, dendrimers, solid lipid nanoparticles, polymeric missiles, and nanosuspensions are. These are the currently being used by the different formulation scientists for the preparation of the or repurposing of the drug delivery system to improve the performance of the dosage form or improve the performance of the drug also. What are the different nano carriers or nanomaterials which are to be used? They are broadly to be classified as a polymeric nanocarriers, lipidic nanocarriers, and miscellaneous one. Among these polymeric nanocarriers, different polymers are to be used for the preparation of nanomaterials are there. That polymers can be the hydrophilic or hydrophobic polymers. In case of polymeric nanospheres and nanocapsules, the drug is to be encapsulated by any means, by different. There are different methods of the preparation of polymeric nanocarriers are there. That is to be called as the bottom top down method and bottom up method is there. These are the two ways of creating the polymeric nanospheres and nanocapsules are there. Whenever there is a matrix type of distribution of the drug into the polymer is to be occur, and if it is exhibiting the size in a nanometric range, they are to be called as the polymeric nanospheres are there. Whenever there is only encapsulation of the drug with the polymeric coat is there, and the particles are exhibiting the size in a nanometric range, we can call it as a polymeric nanocapsule is there. Another form of the polymeric nanocarrier that is called as the polymeric missiles are there. Here in case of this, there is a use of some co-block type of polymers are there, and this co-block type of polymers are having a tendency to orient themselves at a critical micellar concentration, and the hydrophilic end is outside and the hydrophobic core is to be get formed, and the drug substances which are water insoluble in nature that get encapsulated into the hydrophobic core, here one, and in this way there is a encapsulation of the drug is to be occur in a polymeric matrix is that type of system is to be called as the polymeric missiles. Then dendrimers is the another kind of the polymeric system where there is a use of the some branch type of the polymers for the conjugation of the drug or either the encapsulation of the drug by doing some chemical reactions are there that are to be called as the dendrimers are there. Then there are a number of advantages are there with the polymeric nanocarriers are there, like they are hydrophilic in nature, that's why they are not undergoing some phagocytosis type of process is there, and we are able to modulate in terms of co-block polymers. We can easily synthesize the polymeric materials are there, but there is a certain limitations of this polymeric nanocarriers is that all the polymers which are to be used in a manufacturing of nanomaterials are there. There are large molecular weight substances and they are undergoing urinary excretion in them, and that's why they are having some problems of elimination is that. Therefore, to overcome this one, there is some lipidic nanocarriers has been proposed for the preparation of nano-based materials are there. The first type of the system is there, that is called as the solid lipid nanoparticles. Like that of the polymeric nanospheres, instead of polymer, there is a use of solid lipids are there. That solid lipids are imparting some structural integrity to the nanoparticles. But there is again some limitations of the solid lipid nanoparticles is that that will become some leaking on storage. Lipids are unstable in nature. That's why they become somewhat leaky and that creates some stability issues with the solid lipid nanoparticles are there. To avoid this type of the leakiness problem of the solid lipid nanoparticles, the alternate system has been suggested, that is called as the nanostructured lipids are there. In case of solid lipid nanoparticles, the solid lipids are to be melted and the drug is to be loaded into the solid matrix, and then it is subjected for the stabilization in them. Whereas in case of nanostructured lipid carriers, there is a use of the combination of the solid lipids and liquid lipids are there, and that's why there is a structural integrity is to be imparted into the system, and by this way there is a problem of the leakiness of the carrier system that can be avoided. Is that another form of the lipidic nanocarriers, that is the liposomes are there. The liposomes are having these are nothing but the lipid bilayers. The lipids are having some tendency to orient themselves in the form of lipid bilayer, encapsulating the drug into their matrix is that, and thereby increasing the targeting ability of the drug to the specific site is that. Then another type of the lipidic nanocarrier system, that is called as the nano immersion based system. In case of the nano immersion system, there is a use of some oils are there. Depending upon the solubility of the drug in oil, there is selection of the or screening of the oil is to be carried out, and in order to stabilize the system thermodynamically, the surfactant and co-surfactants are to be added, and in this way there is an encapsulation of the drug into the oil phase that is to be achieved, and there is a formation of the nano drug loaded emulsion based system that is to be called as the nano emulsion in them. Then the miscellaneous system consisting of the nano suspension and inorganic nano carrier based nanoparticles are there, that consisting of the gold nano carriers, gold based nano carriers, calcium based, iron oxide base, or some ligand or drug loaded or steel coated inorganic nano carriers are to be used in that. Whereas in case of nano suspension, drug nanoparticles are to be stabilized by using some suspending agents or stabilizing agent after converting into the nanometric size. Ideally, once the drug is to be subjected for the some nanosization process is there, that is somewhat unstable system is there. There is a formation of some agglomerates or lumps will be there. To avoid this one, the stabilizers are to be added in order to maintain the nano size of the particles are there. That is to be called as the nano suspension is there. Now, the distribution of the global nanotechnology drug market in terms of technology based upon the whatever the nano carriers we discussed right now, the major contribution is coming from the nanoparticles are there, and among the nanoparticles, the most of the contribution is coming from the polymeric nanoparticles are there. Then at a second place, there is the liposomes are to be there. And what are the nanotubes and micelles are there that are contributing very little is that. Nowadays there is a nanocrystals are coming into the market, but there are again stability issues with the crystalline drug substances, especially nanocrystals are there, or there is a kind of chances of polymorphism type of events during the nanocrystal preparation. If the drug is having such tendency to undergo polymorphism process is there. Therefore, major contribution in terms of nanotechnology based drug delivery system that is coming from nanoparticles as well as the liposomal based drug delivery systems. Then what is the current scenario is that the nanotechnologies find its application in a targeted drug delivery system is that. Then what advantages we are getting with the nanotechnology based targeted drug delivery system is that, say for example, if drug containing nanoparticles are to be administered, without any target site is done. Then after its administration, the drugs, if it is given in the form of normal drug delivery without any nanoparticles on it, then it will goes to the both healthy cells as well as the disease cells, or what we can say the target cells are there. But once it is given in the form of nanoparticulate based system, and there is if it is a ligands is to be applied, which are having some structural resemblance or affinity toward the only toward the cancerous cells or disease cells are there, and not having a affinity toward the normal cells are there. Then by this way we can modulate the targeted drug delivery system in such a way that it only recognizes the disease cells are there and it is not going to the normal cells. And by this way we are able to increase the efficiency of the drug as well as the carrier system towards the disease cells is there, and we are preventing the toxicity related issues after exposure of the drug to the normal cell. Sorry. And whatever current status of nanotechnology, what we are discussing previously, that application lies here. Nanotechnology in a targeted drug delivery. Whole work in a nanotechnology based drug delivery system going in a US market, that is based upon the targeted drug delivery system for the effective management of the cancers. Now here we are talking about the liposome because this is the first marketed nanotechnology based system is there. In case of liposome, this liposome is first discovered by the one hematologist, British hematologist in 1961. While he is working on some staining of the lipids are there, he found that there is a bilayer orientation of the lipids into the system is to be there, and these self-closing spherical particles that are composed of the natural and synthetic amphiphilic lipid molecules are there. These amphiphilic lipid molecules, once these are added into some specific solvent, they orient themselves and they are having some tendency to accommodate the water insoluble drug into the cavity which is to be formed, whereas the lipid soluble drugs that get encapsulated into the outside layer. Means this is the unique system as compared to the other nanocavity system is that can be useful for the loading of the both the water insoluble as well as the lipid soluble drug substances. Now this liposome based system first reached into the market. This concept of the something is to be there due to the arrangement of the lipids in the form of bilayer as proposed by the that hematologist in 1961 is there. After some period of time, the first liposomal based system is or product is available in the market, that is in the form of Doxil, developed in 1995. It is marketed by Orthobiotech. It is in the US market. It is a liposome PEG doxorubicin hydrochloride is there, in which the doxorubicin is to be loaded into the liposome, and then that has been pegylated to avoid the phagocytotic uptake of the liposomes. Then this liposome is very effective in nature. Liposomal preparation is very effective as compared to the doxorubicin, and it avoided some toxicity issues which are related with the conventional doxorubicin formulation is there, and that's why it is very well recommended for the treatment of the HIV related Kaposi sarcoma. It is also used in a treatment of breast cancer, ovarian cancer, and other solid tumors are there. It is ideally to be administered intravenously every 4 weeks are there, and due to this pegylation of liposome, it becomes somewhat long circulating into the system is that, as it is not easily to be recognized by the endothelial cells in order to undergo some phagocytosis is that, and in this way the half-life of the drug is to be extended, and it is showing some good performance in management of the different kinds of the cancers are there. The Doxil works through the passing through the fenestrations in the vasculature and concentrating at the tumor site, and that's why there is a more effectiveness is to be there, and that leads to the reduced accumulation into the other tissues is to be there. This is the what we can say the beauty of cancer management is that during the cancer there is a the cell geometry gets changed, and that will somewhat become formation of some leaky vasculature is to be there. And being these lipids are present in the form of liposomes are present in the form of some nanocarriers are there that can be easily passed through this leaky vasculature, and that leads to the increased accumulation into the cancerous cells, whereas there is going to changes are to be occur in the vasculature of the normal cell that are not recognized this liposome system, and that's why there is a reduced accumulation into the normal tissues are there. The liposomal system able to deliver the drug at a moderate concentration over a long period of time, that is up to the 54 hours in them. Being it is long circulating in nature, not easily recognized, not undergoing phagocytosis, not removed from the body, that's why it remains into the circulation for longer period of time, and it results into the anti-cancer drugs that are delivered more effectively, and therefore there's a decrease in the side effect as well as the doses frequency of the this doxorubicin. This is the second liposomal based preparation available in market, approved by the FDA in 2004. It is marketed by Sky Pharma. It is a liposomal cytarabine, which is also an anti-cancer agent in the management of the malignant neoplastic meningitis. It is administered intravenously every 2 weeks, and it is showing good results in the management of the cancer. Now coming to the toxicity issues of the nanotechnology based formulation is that due to the nano size, there is a chances that nanoparticles will be present in a as a contaminant into the air, water, or food, or some consumer goods are there, or by any way they are to be ingested into the body or get administered into the body. Say like by inhalation, they are undergoing after the pulmonary exposure, they may undergo inhalation, or through gastrointestinal tract it can undergo ingestion, or through the contact with the skin, they undergo skin absorption. By this way, they are going into the circulation and they are creating some primary effect like toxicity. If it is a very toxic materials are to be used for the conversion into the nanoparticles are there. Then oxidative stress, DNA damage, and inflammation. These are some primary kind of the effects are to be there. Once there is a external administration of the nanoparticle is to be occur in them. Whereas in case of internal nanoparticles from the orthopedic or surgical implants of ears, that is a direct exposure to the cells or tissues is to be there. That is also likely to be have some toxicity issues, oxidative stress, and damage or inflammation into the individual patients in there. Sometimes these primary effects can be translocated into the secondary effects like tissue or organ deposition, secondary toxicity, systemic inflammation, or altered functions of the any specific organ where it get targeted is there. These are some major issues, and that's why every regulatory authority that are giving permission for the nanotechnology based formulation, they are asking for the safety status of the nanomaterials are there, or they are talking about the more studies on a nanotoxicity. Now, while talking about our basmas are there, if you see the basmas are the traditionally nanomedicines are there, they are not undergoing any kind of the metabolism into the body, or they are not creating any active metabolites into the bodies there. That's why they are remain to be non-toxic, because they are not converted into the metabolite. That's why there is no any toxicity will be produced. Whereas the role of these basmas into the treatment is that they are removing some heavy metals which are present into the bodies. Then that is detoxification is to be carried out by the basmas. Yeah. On other side, if you are talking about the emerging nanotechnology based formulation, by any way, either whether they are exposed externally or internally, they are causing some certain kind of the primary effects are there, or if there is a continuous exposure is to be there, then that can be converted into the secondary effects. Then following up some myths about the nanotechnology base material development is that. As we already talked about some limitations of the nanotechnology, it is quite expensive in nature. All these things are there. But while working in a laboratories, what we found that what are the myths about the nanomaterials are there? Like that require the skill to formulate the nanoparticle, need some sophisticated instrumentation facilities, characterization is difficult, and the cost. All these are the some myths about the nanoparticles. While working in our laboratories, what we find, there is a limited skill is required for the generation of nanoparticles are there. If you are having some idea about the properties of the materials are there, drug as well as the material carrier which is to be used for the generation of nanoparticles are there, then you are able to formulate the nanoparticles very easily. There is no need to have some sophisticated facilities for the creation of nanoparticle. That can be possible with the existing laboratory facilities are there. Characterization is somewhat difficult to demonstrate that or to prove that really the nanomaterial is to be present, and cost can be managed by using some easily available excipients or easily available polymers or easily available lipids for the generation of nanoparticles. Now, the critical aspect to demonstrate the what are the particles which are generated, really they are nanoparticles or not, and that is possible by using the some different nano size measurement techniques are there. What are the conventionally used particle sizing techniques are there that are based upon the simple one-dimensional spherical and measuring concept is there, and this openly prefer perfectly adequate for the required application in case of the coarser particles are. But once we are talking about the finer or nanoscale particles are there, these particles are three-dimensional objects are there, and unless they are perfect sphere, for example in case of emulsion or bubble, they cannot be fully described by single dimension or conventional microscopic type of techniques are there. For that purpose, we need some three-dimensional object measurement technique to demonstrate whether exactly the material which is to be obtained, it is having a nano size or not is that. For this nano size measurement, the commonly used technique nowadays available with us, that is a dynamic light scattering technique is there, where in case of this, the nanoparticle suspension that is to be kept in a sample cell, there is a laser beam is to be applied, and due to the these nanoparticles which are present in a suspended form is done, there is a this laser beam get diffracted is done or scattered is done, and that scattered light is to be intensity of the scattered light that is to be recorded at a by using some detectors are there in terms of the scattering intensity, and that scattering intensity has been converted in the form of the average particle size on. Ideally, if it is a nanoparticulate system and if it is present in a suspension form, then all the particles which are in a three-dimensional, which are having a three-dimensional structure in them, they are in a Brownian motion and they are always coming in a path of the laser beam is there, and due to this there is a scattering of the light is to be occur, and the what is the scattering intensity is to be done, that is detected by the detector. Ideally they are located at either at a 90° or at the 170°C, and then this scattering intensity is to be converted in the form of the average particle size by using some signal processors, by using some softwares are there. Now we'll see if it is a smaller the particles are there, then what will be the case is that if the smaller the particles, they are moving around, and whenever this laser beam is to be passed through the sample holder is there is a scattering is to be occur, and if you see this scattering will be the very small in size is there, or the intensity is very big as compared to the larger particle. If it is the larger particle, then this scattering intensity will be the more is there, and that can be giving you the size in a nanometric range, whereas in case of larger particles, the size distribution data is going in the outside of the nanometric range is there. Now, once you are coming comparing the intensity terms is that which will be the ideal way of measuring the particle size in terms of the nanoparticles are there. Say for example, if you are going for the number of counting is that to determine the particle size, and for a sample consisting of equal number of particles with diameter of 5 nanometer and 50 nanometer is that. Then in terms of numbers, if you see, there will be the equal numbers of the particles will be there, which are having particles of 5 nanometer and 50 nanometer. Then you are unable to differentiate between which is the exact particle size of the dispersion system or population of the particles are there. In terms of the weighted distribution, gives equal weighting to the both types of the particles are there, whereas the intensity weight distribution has the signal 1 million higher for the coarser particles. Just previously we discussed, if it is a coarser particles, the intensity will be more. In case of the nanoparticles, the intensity will be more. That can be clearly indicated. In terms of volume, if you see, it is in between the this number and intensity. But if it is a coarser particle, that is having the intensity percentage of intensity is more as compared to the nanomaterials. But while studying the distribution parameters are there, ideally the nanoparticles which are to be used for the preparation of dosage form is there, that should have the uniform distribution of the particles are there. But it is not always possible to develop the particles which are having the normal distribution of the sizes. Then there are 3 ways of the determining the particle size is there. Mean particle size, that is the average size of the population, median size of the population where 50% of the population is below or above is there, and mode size with the highest frequency is there. If it is a case, the polymeric system, nanoparticulate system is consisting of the uniform distribution of particle, then in that case, either you are measuring the mean, or either you are measuring the median, or either you are measuring the mode is there. That will not matter. Every time you are getting the exact particle size. But this is not possible in case of the nanotechnology based product development is that there is a normal distribution will not be there. There is a chances of formation of the bimodal distribution or multimodal distribution is there. Then in that case, if you are talking about the median is there, then what is the median? 50% distribution is there. Then what will be the case is that here in that case, you are getting the particle size which is represented by only the 1% of the particles are there. Whereas in case of mode, say for example, if you are having the such particles distribution, 49% are having one size and 51% is having remaining 51% are having some size in them. Then if you are measuring the mode, then you are only counting the 51% particles are there, which is wrongly to be taken as a highest frequency is there. But if you see mean or average of the population is there, then you are actually to be measuring the average of the both 49 plus 51, that will give the 50%, and that give the exact idea about the size distribution data. Distribution statistic is very important to demonstrate the nano size of the material is there. Then another important aspect in a nanoparticle characterization, that is a zeta potential measurement, that is to demonstrate the stability of the system is there, or to measure the some charge which is to be present on a system is there, nanoparticulate based system is there. It is the fundamental parameter known to affect the dispersion stability. And once you are talking about the conversion of the nanoparticles into the dosage form is that it should have the sufficient zeta potential value to have some desired stability on storage is that. Ideally, the zeta potential value should be in between plus 30 to minus 30 to demonstrate the good dispersion stability is that. Then how to measure this zeta potential? That zeta potential can be measured by using the charge on a surface is there. The charge or zeta potential of particle or molecule is determined by measuring the velocity while they are moving due to the electrophoresis is there. There is 2 electrodes are to be provided. Just a minute, if it is possible, then I will show you. Yes. Here, the nanoparticle dispersion is to be filled between these 2 electrodes are there. One is anode and cathode. And due to the electrophoretic mobility of these particles which are enclosed into the cell is there, This cell is to be called as the zeta cell is there. And here, if you are able to recognize, these are nothing but some electrodes are to be provided on both sides of it. That electrodes are the particles which are having an opposite charge that are moving toward the electrode, and that are giving you the zeta potential values there. The speed they move is proportional to the field strength, and the zeta potential is there. If you know the field strength, and we simply measure the speed of movement using a laser Doppler electrophoresis, or that is also to be called as the zeta meter is there, and we are able to demonstrate the stability of the nano suspension or nanoparticulate based system. Following are some applications of nanomaterials in a drug delivery system. There are a lot of reports are available about the application of nanomaterials in intranasal drug delivery, transdermal drug delivery, parenteral drug delivery, drug targeting, vaccine delivery, and pulmonary drug delivery is there. This is our some little bit research work on nanoemulsion for CNS targeting. We have developed one nanoemulsion of saquinavir mesylate for nasal delivery, and the objective of the study was to assess and enhance the targeting of saquinavir mesylate to the CNS after its intranasal administration. We have prepared the nanoemulsion by using a spontaneous emulsification technique. While talking about the myths about the nano preparation formulation, we prepared this nanoemulsion by using a simple vertex mixer available in a laboratory by using the oil phase as a capmol MCM, that is a glyceryl monocaprylate, surfactant 280 and co-surfactant PEG 400 is that. Then after preparation of the nanoemulsion, we subjected it for the characterization purpose. The average global size is to be found 170 nanometer with a PDI value, that is indicator of the normal distribution of the particle is there, that is 0.078. It should be below 0.5 to demonstrate the normal distribution of the particles are there. The zeta potential values falls between falls to be minus 10.3, which is between 10 to plus 30 to minus 30 is there. The refractive index is 1.41, which is similar to the refractive index of water. That is the one of the means of demonstrating that what is the resultant system is to be formed. That is not the microemulsion, it is a nanoemulsion system is that. Then we put this formulation or subject this formulation for ciliotoxicity study, and after exposure to the very sensitive nasal mucosa, this is the intact nasal mucosa after exposure toward the drug loaded nano emulsion is there. Same thing is observed in case of after exposure to the phosphate buffer solution pH 6.4. Once it is subjected for the exposure to the isopropyl alcohol, there's a damage to the nasal mucosa has been observed. That clearly indicating what is the nanoemulsion we have prepared in our laboratory that is non-toxic to the sensitive nasal mucosa is there. Then in order to demonstrate the CNS distribution study, after administration, we estimated the amount of the drug after administration in the form of pure drug suspension and after the administration of nanoemulsion in that. And there is a significant concentration of drug is to be observed. In case of nanoemulsion, which is quite greater, or 40 folds increase in the concentration is to be occur into the brain, as well as the similar enhancement is to be occur in a blood is to be there. That is clearly indicating that by preparing the nanoemulsion of this saquinavir mesylate, we are able to target the drug to the central nervous system is there in an effective way. This is the proof of concept. After the administration, initially there is no any amount of the drug is present into the CNS of the drug animal is there. After half an hour, there is a significant amount of the drug is to be achieved in a CNS, and immediately after that there is a excretion of the drug is to be also started. As indicated in amount of the drug accumulated into the kidney of rabbit, which are used for the gamma scintigraphy study, that indicating that there is no any toxicity, accumulation related toxicity will be possible by this type of the drug delivery system is there. And in this way, we are effectively targeted the drug to the central nervous system is there. To sum up with the nanotechnology based drug delivery system, the development of particles that are nanoscale has created a great opportunities in a development of the improved drug delivery system. By doing this one, we can improve the drug delivery system, or we can overcome some limitations of the conventional or novel drug delivery systems which are currently available with us. Nanomaterials have increased the surface area, nanoscale effects, hence they are used as a promising tool for the advanced drug as well as the gene delivery, biomedical imaging, and diagnostic biosensor applications are there. Hence, with the help of nanomedicine, early detection as well as the prevention and improved diagnosis, proper treatment, and follow-up of the disease is possible is that. If you have any question regarding this, you can put in a chat box.
First of all, I would like to thank you very much, sir, for sharing valuable information with us. It was really informative session. And sir, you have explained many important points. Some I would like to mention over here, like progress in nanotechnology, advantages and disadvantages of nanotechnology, importance of nanoparticles, general properties of nanoscale particles, various nanocarriers currently being used. You have also explained nanotechnology in targeted drug delivery system, different liposomes which are used in the cancer treatment, then nanotoxicity, and also you have explained well regarding nano-size measurement, distribution statistics, Zetam potential measurement, and applications and characterization. So I'm sure definitely this information will be useful for our students as well as all the participants present over here. Definitely we will utilize this information. Once again, thank you very much, sir, for this wonderful session. Now, I would like to request Ms. Namita Bhosle, ma'am, please proceed with the question answer session. Thank you so much, sir.
Thank you so much, Shweta ma'am. We shall proceed with the question answer session. The participants are welcome to put forward their queries, if any, in the chat box. Dear participants, you all are requested to put forward your queries in the chat box. I think sir has wonderfully covered the session, so no one has any query or doubt. So, sir, shall we proceed with the next session? Okay, we will wait for 1 minute.
Dear participants, if you have any queries, you can put in the chat box.
Okay, once again, thank you so much, sir, for the remarkable session. No one has query, so credit goes to you, sir. Thank you so much.
Thank you.
Thank
you. If you have any question, you can write mail to me. If you're having some work to be done, because we are having all facilities related to the nanotechnology based formulation development as well as the characterization in the R.C. Patel Institute of Pharmaceutical Education and Research. You are very welcome at any time, and we are providing all type of guidance for formulation development as well as the characterization purpose. I'm thankful to organizers for giving me an opportunity to share my views on this nanotechnology based drug delivery system. Thank you very much.
Thank you, Majan sir. Thank you for your presence.
Thank you.
Good day.
Okay, thank you so much. Now let's take up again with our next technical session, which is based on recent advancement in novel drug delivery system. To chair the session, I would like to request Mr. Mayuresh Redikar sir, Assistant Professor, Department of Pharmaceutics, to take the opportunity to introduce our today's resource person for the second technical session, Honorable Dr. Bhushan Rane sir. Sir, please.
Am I audible, ma'am? Hello, am I audible?
Yes, sir, you are audible.
Okay. Thank you so much, ma'am, for this wonderful opportunity. And it's my great privilege to present the profile of our eminent speaker of today's program. First of all, good afternoon to all. Honorable Dr. Bhushan Rajendra Rani sir is currently working as an associate professor and head of pharmaceutics department at Sri D.D. Vispute College of Pharmacy and Research Center, Panvel. He has more than 13 years of teaching and research experience. Sir is approved by University of Mumbai and North Maharashtra University, Jalgaon, as an associate professor and postgraduate teacher in pharmaceutics and quality assurance subject. He gave his guidance to more than 42 MPharm students. He pursued his degree of PhD from Jodhpur National University, Jodhpur in 2013 and completed his MPharm from Annamalai University, Chidambaram, Tamil Nadu in 2007. Sir has initiated his research in MPharm on process validation of theophylline sustained release tablet, and further in PhD he studied floating microcarrier for drug delivery of anti-diabetic drugs. Sir has received many research grants from industries as well as from government schemes as the principal investigator and co-investigators in various research projects with an amount of more than 14 lakhs. Sir has received grants for MODROP scheme. Sir also received various minor research grants for the research topics entitled Formulation of Nanoparticulate System, Development of Herbal Formulations, Formulations of Novel Ultra-Deformable Vesicular System, and Formulation and Evaluation of Novel Chocolate Formulation of Herbal Immunomodulators. He is appointed as a member of Board of Studies, BTech Cosmetics at North Maharashtra University, Jalgaon, member of Local Inspection Committee at University of Mumbai and North Maharashtra University, Jalgaon, appointed as chairman and paper setter at North Maharashtra University, Jalgaon for undergraduate and postgraduate subject. He also appointed as a quality manager at ASR CTL, New Panvel, and successfully faced NABL audit. Sir was also invited as a guest resource person, speaker, and judge in many national level scientific programs. Sir is in the reviewer panel of various journals, including Journal of Pharmaceutical Technology and Drug Development, Journal of Asian Journal of Pharmaceutics, Bentham publication, Journal of Drug Delivery Science and Technology, Journal of Current Drug Delivery and Current Analytical Chemistry, IGI Global Publishers, Online Books, Taylor and Francis Group, Apple Academic Press Publishers, Online Books, Taylor and Francis Groups. Sir has published 3 books, including A Glimpse of Novel Vesicular Drug Delivery System, Pharmaceutical Engineering, and Practical Book of Pharmaceutical Engineering, published by Nirali Publishers, Pune. Sir also had 8 book chapters in his account and more than 75 papers published in national and international journals and presented 19 papers and posters in various conferences. With this brief introduction of Honorable Dr. Bhushan Rane sir, I would like to request Dr. Rane sir to proceed with second scientific session entitled as Recent Advancement in Novel Drug Delivery System. Over to you, sir.
Thank you, sir. Shall I share my screen?
Yes, sir.
Is it visible, sir?
Yes, Bhushan sir.
Okay. So, a very warm good afternoon to all. All those present over here. Is it visible, sir, clearly?
Yes, sir.
Okay. So, I will start with that second session. Recent advancement in the novel drug delivery systems. Thank you that faculty member for my brief introduction. So no need to explain that other details. So just move to the next part. The lot of questions are there in the every researcher's minds that what type of drug delivery systems are available and why we have to prepare such a type of systems. So basically we start with first the basic thing, the drug delivery system. That is the number of approaches that formulation technologies and the systems, those are available for the researchers to transport an active pharmaceutical ingredient into the blood compartment, into the systemic circulation or the body. And the basic objective behind this preparation of this dosage form is that to just release the drug from the dosage form into the blood compartment, and thus automatically the drug will show its therapeutic response or the effect. Now the question is that on what aspects or what will be the basic criteria behind the development of drug delivery systems. So first one is the route of entry and second one is the dosage form. Dosage form, that is a solid, liquid, semi-solid, and the route of entry is by oral route, topical route, and the parenteral route, many more. In the first year reform, every students learn about that, the route of entry and the different types of dosage forms. So we'll move to the next slide. Nowadays, broadly, the two types of drug delivery systems are considered. The one is a conventional drug delivery system, that is your tablet, capsule, normal, conventional, without any sustenance polymer. And the second one is the novel drug delivery system. So whenever any students or any researchers admitted for PG and the PhD program, at that time in his mind or her mind, the basic thing is that I have to work on the novel system. So question is that what is mean by novel? That is a main question or big question mark is there in front of the every researcher. So what is novel? So we just highlight on that aspects of novel drug system today. So first of all, before going to start the novel delivery system, you have to always learn what is meant by conventional delivery system and why these conventional delivery systems have some drawbacks. And that's why the researchers started to invent or develop the novel drug delivery system. Now, basically that conventional drug delivery system in which the active drug is released immediately after its administration within a few minutes. Right? Now the question is that what type of problems are there with the old concept, old or traditional conventional delivery system, and that's why researchers just pull the concept of novel drug delivery system. So the drawbacks associated with the conventional drug delivery system is in the case of conventional delivery system, the release of drug, it is uncontrolled, right? The some sort of adverse effects are more, side effect of drug is there, the patient non-compliance is there in some cases of dosage form, as well as it is very difficult to maintain that therapeutic window level in the blood. Later on, in that conventional systems, the dosing frequency is more, means the patient have to take the dosage form in morning, afternoon, evening, and at night. But nowadays, because of that novel delivery system, you can reduce the dosing frequency. So per day, per month, per week, or per year, the patient have to take the dosage form only once. Depend on that what type of dosage form it will be there. Again, some drawbacks are there, that many times that conventional dosage form deliver the drug at high concentration, and which leads to the certain sort of side effects, as well as the in cases of that cancerous therapy, because of that conventional delivery system, the drug will target to the normal cells also, so which will lead to the other issues. So these are the drawbacks of conventional drug delivery system, and which will be overcome in the novel drug delivery systems. Now we'll move to the next part, that is the novel drug delivery systems or the recent advancement or new technologies in the NDDS. So it will be encouraged by the old concept of magic bullet, that is, which is to be proposed by a great scientist, Paul Enrich. That is basically old moiety is there, the drug is the old, but you have to just wear with the new cloths or the carrier material, and that's why it will show, or its control release will be there, sustained release will be there, targeted release will be there, and that is nothing but the new technologies with the novel drug delivery systems. That is, you have to take. I will suggest that every researchers, no need to go for new drugs. You just take the old drug. The drawbacks of that old drugs, that will be overcome by applying the basic novel approaches, and that is nothing but the new technologies of the NDDS. Now, the new drug delivery system development is largely based on what type of concepts? The concept is that the researchers, they will think on that the drug will be released in the sustained manner at the predetermined rate. Second approach is that basically the dosage form should have to follow the zero order kinetics, so that we will maintain that constant drug level in the body throughout the desired period of time. As well as by developing the novel systems, we will promote the therapeutic effect of drug at the degradation of drug will be minimized. Next, we can minimize the toxic effects of the drug by increasing the amount and time of drug in the close contact with the target cells, that is cancerous, tumorous cells, as well as we can reduce the drug exposure towards the non-target cells, that is healthy cells. No need to expose our drug with the that cells. So we can minimize by the using specific carriers, and through that specific carriers, we can target to specific cells only. Now, minimizing the drug concentration, the drug degradation and the loss of drug by just formulating a particular novel systems in which the drug is encapsulated into that polymer matrix, and that's why the drug will be protected from that specified physiological environment of the body fluids. As well as we can increase the bioavailability of the drug by different approaches. That is, we can increase the solubility of the drug by different approaches are available, such as solid dispersion, micronization, homogenization, right? Emulsion, suspension, whatever it may be. And ultimately, because of that, the rate of absorption will be increased, and which will lead to the increased bioavailability into the blood compartments. Now, next, that is therapeutic and clinical advantages of novel drug systems. Many advantages are there. So you can treat the chronic illnesses, that is cancer, asthma, arthritis, by developing and just by giving such a dosage forms to the patients, as well as we can prevent the hepatic first pass metabolism of the drugs, though will get metabolized into the body after oral administration. We can improve the outcomes of the treatments. We can reduce the adverse effects of the drugs by applying different approaches, as well as the optimization and the duration of the action of the drug also will be there improved. We can decrease the dosing frequency, which I already discussed, that as the 2 to 3 times that you have to administer the tablet, instead of that, if we just convert that system into the sustained release or matrix type system, that time ultimately the dosing frequency will be reduced. Because of this development of novel drug systems, the efficacy of the drug is also increased. By this NDDS or the targeted drug system, more specifically, we can target the drug at specific site, to the specific organ, to specific tissue, or the specific part of the body. And at the end, ultimately, the bioavailability of the drug will be increased. Now, the viable treatments for previously incurable diseases through the gene therapy, it is also one of the approach which is available with us, so we can develop. Now next, in certain cases of prophylactic applications, this NDD role. Now NDDS, because of that novel dosage forms, the patient compliance is there, patient feel happy when it will taken, because of the dosing reduction in the dosing frequency. Per day you have to take only 1 tablet instead of taking more tablets into different time intervals. As well as we can improve the stability of hydrophobic drugs. Now many students, they are confused with the hydrophobic and the non-polar system. Hydrophobic and non-polar both are different actually. All the non-polar systems and the hydrophobic systems, they are classified, they will be defined in a different manner. Likewise in polar and hydrophilic. Polar is, that is, it will have the 2 charges when it is get prepared at aqueous solution, that is partial positive and negative charge. While hydrophilic, that is, they always have the some sort of affinity towards the polar solvents, right? So all the polar molecules are hydrophilic. Likewise, all the non-polar molecules are hydrophobic. Next, but always there is some sort of advantages. Again, with some sort of advantages, some sort of limitations are with the NDDS. That is, if we some sort that particular dosage form, and if the system is fails to release the drug, or some sort of other options are there because of the patient to patient vary, and that time it may happen that dose dumping is there, the over amount of drug which will be released from the system, and which leads to the toxicity. Some other limitations are there, that is large physical size of dosage unit. If you have to prepare the sustained release tablet, and in case the dose of the drug is 500 milligram, and at that time, if the researcher have to prepare the tablet in the ratio of 1:3 to sustain the dosage form more than 24 hours, that time automatically that average weight of tablet will be more than 2 gram, and it is not feasible for the human being that we can unable to swallow the tablets of average weight of 2 gram. Often that low bioavailability is observed in some cases. Again, some limitations are there, so we'll not look into that limitations, because we have to focus on what are the advantages of the NDDS, because every researcher want to prepare the novel system. So first of all, I will highlight the basic applications of the NDDS in the pharma field. That is, you can target the drug to the brain via nasal route, nose. Second approach is mucosal drug delivery system, that is mucoadhesive systems are there into that mouth cavity, stomach, intestine, vaginal cavity. Third approach is pulmonary drug delivery systems. This approach, it will be there. Basically, all of you aware about that coronavirus that will target to the lungs, that pulmonary tract. So it will be an opportunity for the researchers that you can just take these pulmonary drug systems, find out the basic drugs, those will be there for that respiratory tract applications, and you can develop the novel drug delivery system. As well as skin drug delivery. All are aware about that. Nowadays, the temperature, because of that global warming, the temperature will be 45 to 48 degrees Celsius in that summer seasons. And many patients, many individuals suffer from that tanning, skin tanning problems. So this will be an opportunity for the cosmetic industry that you will develop the novel concept that is transposome, neosomes, that nanogels, in which you can incorporate the various herbal drug, the extract of herbal drugs, and you just prepare that nanogels, that topical applications, topical dosage form, those will be applied topically in order to treat that skin problems or the skin issues. Ultimately, day by day, that global warming will be there, and it will lead to that develop in future that skin cancer patients will be more and more. So it is an opportunity for the researchers to prepare the formations. Those are related with the transposomes and the neosomes or the various vesicular approaches. The next application is anti-cancer drug delivery is there. So that paclitaxel, all may aware about it, that paclitaxel concept. Peptides and protein drug delivery, most important, proteins and peptides get degraded into stomach when they are administered by oral route. So it is a very big task for the researchers to develop a dosage form in which you can incorporate or add the proteins and peptides as they contain CONH2 group, and they will be that some sort of other molecules, that excipients, that complexations will be there, degradation of the product will be there easily in that acidic environment. So it is a big task for the researcher to develop the proteins and peptides novel dosage form. This one example is there, insulin. Okay. Next, through these novel delivery systems, we can deliver the genes also, mutations of the genes, changes, and more important, nutraceuticals. Nowadays, in that last 10 years, the nutraceuticals will capture the biggest market throughout the globe, as during these COVID situations, every individuals aware about that prevention is better than cure. So through these nutraceuticals, immunomodulators, you have to maintain our immunity. So I will suggest the students of PG program, those are present over here, try to prepare some of the nutraceutical formulations and evaluate it. And ultimately, the different schemes are there in the every district. The MCD there, that is Maharashtra Center for Entrepreneurship Development is there, through which you can just start your small scale industry also for your PG dissertation work. If you just go with that nutraceutical formulation, so you can launch your own product into the market. So don't try to be a that worker, or don't go for that salary. Try to be an entrepreneur. That is my suggestion. Next, more important, those are try to start their own small scale industry initially. So what will be the industry scenario nowadays into that global market? That NDDS is expected to be a US dollar 320 billion up to the end of 2021. This increase in the NDDS market is only because of the increased requirement which will be shown by the NDDS with its improved efficacy, safety, and biorecognition of the drugs and its site specificity for the targeted drug delivery. Move on to the next. For the pharmaceutical industry, NDDS have the potential to extend the patent life. Now question is that how to extend the patent life. So in that, previously I discussed that old drug with the new cloths, Paul enriched concept. So we will move to that next polymers developments, next dosage form developments. You can apply it for that new patents on the prepared dosage form with the novel polymers and novel systems. Through this, we can improve the possibility of success of many existing and the new therapeutic agents, for which lot of issues are there. That is basically that solubility problems, that is class 2 drugs, low solubility and high permeability. Ultimately, because of that low solubility, the rate of absorption is less, lower amount of molecular solution is available for that absorption site, and ultimately which leads to the less bioavailability. As I discussed, that protein and peptide delivery problems, as proteins and peptides get degraded into that acidic environments, and as well as lot of issues are there with the drugs, so that can be sorted out by this NDDS, the potential of the NDDS. Broadly, that novel drug systems are classified as per according to that vesicular systems. Number of approaches are available, that is liposome, neosome, transferosome, virosome, discosomes, cubosomes. Multiple vesicular systems are there. So I will highlight on to that particular approaches in that next slides. As per according to the route of administrations, the NDDS are classified as oral, that is sustenance tablets, transdermal, that is nanogel. Next one is that based on that mechanism of drug release, if the drug is released is depend on that physical mechanisms, that is osmosis, diffusion, dissolution, matrix type. And if the drug is released by biochemical mechanisms, that is monoclonal antibodies, gene therapy is there, vector systems are there, polymer drug adducts are there, or again that liposomes, through that phospholipid core the drug will be released, and because of that liposomes the permeability will be enhanced. The next classification, the type of the NDDS, which is based on the drug release, the control behavior of the drug release, that is matrix diffusion control types, dissolution control matrix types, dissolution and diffusion both systems, water penetration, that osmotic pressure, it will always play a role, that is osmotic pumps are there in which osmotic pressure, osmogen is added into it, and that it will create osmotic pressure to release the certain amount of drug into the control manner through the system. Next one is a chemically controlled novel drug system, that is prodrug concept, that will be get metabolized into the body, and later on it will show its effect. Hydrogels are there, organogels are there, as that hydrogel is when that aqueous system is there, aqueous solvent systems are used to prepare the gel, it will become a hydrogel, and if the organic solvents are used, it will become a organogel, that is ethanol. Next approach is that classification is ion exchange resins control systems, that is cation and anionic exchange resins can be used, and so when they get administered they will form a complex with the cations and anions, those are present in our body, and drug will be released into the body compartments. And last classification, that is control release, sustenance and the pulsatile systems. Now through these various approaches, through these various drug delivery systems, the method is specifically based on that we must have to achieve the significant effect of drug, and thus it will show its highest efficacy, the slow progress in the treatment of severe diseases, growing need for the multidisciplinary approach for the delivery of certain therapeutics. Currently, Lot of that novel approaches are available, but the work is going on that targeted drug delivery system specifically for the treatment of cancer tumors. As you know that nowadays that cancer patients, the numbers will be get increased, which is only because of some sort of that foods, cosmetics, those are used by the individuals, and that as well as that carbonated beverages, that as that farmers use the more and more amount of pesticides, the spreading of pesticides is there, the air pollution is there, the food is contaminated with the pesticides, and when that pesticides, when they enter into our body, they will directly affect our cell division process. And when they affect on our cell division process, normally when the cell division is there, that 50-50 proportion separated out and that two cells will be formed. But when that certain allergens, that is pesticides, when they affect on that particular normal process of cell division, what will happen? Abnormal cells are there. That is not the 50-50, but 40-60, 30-70. So one cell will become a more, some tiny cells are there, some abnormal cells will be grow, and because of why? The cancerous cells will grow. Initially, if that immunity of the human being is more and more, these abnormal cells will be destroyed by our self-immunity mechanism. But when the more and more amount of that pesticides, the allergens, when they attack our body, that time more and more of cells will be there, abnormal cells will be grow, and that time our immune system will not be that much level of affinity to destroy such abnormal cells. That time we must have to take the help of certain medicines. So here the targeted delivery systems can be achieved by these novel approaches by using suitable carriers to target the drug. Now, whenever we have to prepare any drug delivery systems, that time in every researcher's minds, three basic concepts must be there before formulating any dosage form. That is, what type of drug formulation I want to prepare and why. Second thing, is it medical device or dosage form that I have selected, which is based on what type of technology, and when the system goes inside, how the drug will be get released. That is the third, that is mechanism of the drug release. Lot of drug release systems are available. Those are based on these three aspects. First one is that carrier-based drug delivery systems, in which number of carriers are used in order to target the specific cells, organs, in order to increase the permeability of the drug. That second approach, that is transdermal drug delivery system, that is transferosomes. Third one is mucoadhesive drug delivery system, where you have to target the drug release into that specific region. So all of you knows, if you have to target the drug, or you have to release the drug only and only in the stomach, that time you have to prepare that mucoadhesive tablet. So when that tablet is administered, it will stick to that stomach internal mucosa, and the drug will be released into the acidic environment only. So that is mucoadhesive drug delivery systems. In case of mucoadhesive vaginal systems, that time our system will be added to the mucus of the vagina, inner layer of the vagina. Next one is that osmotically controlled, that is osmotic pumps are available. And last one is that drug devices, that is ocuserts, inserts are there, other devices are there, that is insulin pens are there. Now, different drug delivery carriers are available. They will be categorized as per according to their nature as hydrophobic and hydrophilic, as well as the selection of these carriers totally based on what type of drug, the drug physicochemical properties, that is its pK value, its half-life, its molecular weight, its log P value, its solubility. According to that, the carriers will be selected, as well as the organ, the targeted organ where you want to target your drug, the specific semi-permeable membrane, that receptors available. According to that, the carriers will be selected. Different advantages of this targeted delivery system is the carriers into the targeted delivery systems and the control release systems, as we can maintain the therapeutic window of the drug. Second advantage is some carriers are biodegradable, non-immunogenic, biocompatible, as they are get administered. So because of our body metabolic systems, they will be get biodegraded, and ultimately they will be excreted out from the body. So just think on it, if the carriers or the polymers, if they are not biodegradable, if they are not metabolized by our body systems, what will happen? After certain period of time, the years and months, every human being, they must have to go for operations, and they have to just remove that tumor-like structure of that polymers. Those are accumulated into the body. Means normally, if that polymer, that likewise our HPMC, ethyl cellulose, Utrazit S100, if they are not degraded, what will happen? They will accumulate into the body, and ultimately, day by day, the size of that polymers will be get increased, and the tumor-like structure will be developed into the body, and which will show the disturbances into the normal physiological functions of the body system. So after a particular period of time, the patients have to operate and they have to remove that tumors. These drug delivery carriers, they are very useful when the drugs, they will show the first pass metabolism or hepatic metabolism. So you can avoid the drug contact with that stomach fluid. When the drug will be get degraded, you just protect that drug, and just it will be passed from the stomach environment and goes into the intestine, and when it goes to the intestine, it will show its release. One basic example is for that acid sensitive drug is entry coated tablets. When the drug is coated with the Utrazit S100, as Utrazit S100 is insoluble in stomach fluid, that is pH of 1.2, so the tablet remain as it is intact, and after 2 hours of our gastric emptying time, it will be passage and goes into the intestine, and into the upper part of intestine, our tablet, that polymer coat of Utrazit S100 start to dissolve at pH 5.5, and the drug release will be there. Next, that minimal amount of drug release is also there through these various carriers, as they will form a coat around our drug environment. Now, this is a basic about why we have to develop the novel drug delivery systems. When we have to develop the novel drug delivery systems, what type of points that we have to check, investigate, or the researchers must have to think on it before formulating the novel drug delivery systems, and how to select the drug. That previously, if the drugs have problem with the solubility, that time you can work on the enhancement of solubility. Previously, the old drug which is having the issue with the permeability, then you can prepare the liposomes, neosomes, transferosomes. If there is an issue with the bioavailability, they can use the proper, you just apply the basically the novel drug system approach onto that drug to improve some sort of the bioavailability. So here, lot of approaches are there. That is micelles, liposomes, neosomes, ethosomes, transferosomes, virosomes, cochleates are there, cubosomes are there, e-liposomes, cyclodesting for the enhancement of solubility, dendrimers, nanoparticles. Into that nanoparticles, Previously, Mahajan sir already highlighted on the topic that what are the nanoparticles, the different dosage forms, immunoconjugates, that antigen antibody complex therapy, microspheres, resealed erythrocytes, more specifically useful in the sickle cell anemia. Multiple vesicular approaches are available. So here I unable to go to that next slide of that nanoparticle. Here I just hyperlink that classes of nanoparticles. So lastly, I will reveal that classes. So just move to first point, that micellar phenomena. During preparation of emulsions, suspensions, all you aware about the concept of micelles, that surfactants, when that surfactants are added into that aqueous environment, according to that, their nature of hydrophilic head is there and hydrophobic tail is there, they will orient self and they will form a particular self-assembly mechanism. So within that micelles, the drug will be get enclosed into that lipophilic. So if there is an issue with our lipoidal molecules, that time the drug molecules will be get encapsulated inside the core of the hydrophobic region of the surfactants. So this is that micelles. Both the example is that estradiol, the trade name is Istasorb, which is a FDA approved micelle formulation, which is topically for the treatment of vasomotor symptoms of menopause. Some other systems are also there. They are under the trials nowadays. Now, liposomes. It is a first approach or novel approach which categorize under the class of vesicular systems. So here I just discuss the different approaches and how or what type of drugs will be get encapsulated into the liposomes. So just see, the basically phospholipids are there. Phospholipids are the molecules, they are chemicals, biochemically. They have one hydrophilic head and the hydrophobic tail is there. When these phospholipids are added into the aqueous environment, that time what will happen? They will form a double layer phospholipid. Automatically, they will form a stack-like structure, just like you're playing cards. One above the one, the layer will be formed of phospholipids. Now, when we go for the next stage of evaporation of that aqueous environment, that time the dried film is formed. And when this dried film is again hydrated with the phosphate buffer solution of aqueous environment, that time this stack-like structure of phospholipids, layer by layer, is swelled. And after that, this swell stacks, when they undergo the sonication process, the vesicles will be formed, the close assembly is there. The molecules will be in the close vicinity with each other. And during this process, the drug molecule which is available into that external aqueous environment, it will be get enclosed inside the core. So here in that liposomes, the hydrophilic, hydrophobic, both the type of drugs will be get encapsulated. Even the DNA molecules can be encapsulated. Hormones and peptides, they will be adsorbed onto the surfaces. Even onto that phospholipid surfaces, that head region, you can attach some sort of carriers molecules. And so these carriers have ability to carry your whole liposome molecules to the target region. These carriers will bind to the receptors. Phospholipid core will be get open, drug will be released, goes into the cell, and it will show itself sort of its activity according to the nature of our drug molecule. So this is basic application of liposomes. So the issue with all the drugs with that permeability, class 3 drugs where that issue is there, class 4 drugs where the solubility and permeability also that issue. So we can try to prepare the liposomes in order to solve that permeability issue. If the permeability is the rate limiting step, Now here I discuss some approaches. Those are fall in the category of vesicles. So I discuss that liposome. Next one is that ethosomes, where it is similar to that liposomes. Just here, instead of that aqueous, we have to add the ethanol for the increased flexibility of the system, so that it can easily crosses that our skin barriers, that semi-permeable membranes. So that time they are termed as a ethosomes. That E stands for ethanol. Virosomes, that word itself reveals all the information about that vesicles, that virus, that is it something related to the virus. So liposomes are there which will able to carry the spike proteins of virus onto their surfaces. That virus will be there onto the surface of the liposomes. E liposomes, that E stands for emulsion droplets. That emulsion nanodroplets are encapsulated into the liposome. That time it will be termed as a E liposome. Next one, that is magnetoliposomes, in which that magnetic nanoparticles, if they are encapsulated into the liposomes, that time this novel approach will be termed as a magnetoliposomes. Next approach is the neosomes. So in order to maintain the stability, all are aware that in the liposome phospholipids are used, and all know, understand that if that fatty materials are there, phospholipids are there, they are always undergo that auto oxidation process. And because of auto oxidation of phospholipid, that is fats and oils, like our ghee, it will become rancid like our coconut oil, and our system will become unstable. So to give the stability to our system, the non-ionic surfactants are used, and that's why that surfactants of non-ionic, it will be termed as niosomes. NIO, that stands for the non-ionic surfactant. Next one is that proneosomes. All the students are aware the concept of prodrug. That is a primary stage, that when the prodrug is administered into the body, it is get metabolized, and after that metabolism, the drug will be there, and it is available for the some sort of therapeutic response. So likewise here, the proneosomes, that is a in that powder form, neosomal dispersion, when come in contact with the body fluid, it will form a neosomes. So that is the proneosomes concept is there. All the students or researchers, they many times observe that physician, when they can administer some sort of the dosage form, in that ampoules only powder formation is there, and adjacent to it one water for injection is available. So at the time of administration, both the systems are mixed. So as the issue is that when the stability problem with our drug is there, that time we have to try these proneosomes. The spray dried formulations are also there, freeze dried products are also there. Next approach is cubosomes. That word itself indicate the system looks like a cubes. That honeycomb structure like system is available, in which within that combs, that cubes, the drug is encapsulated, and whenever that one by one cube is come out from that whole structure, the drug will be released or come out, which is available for the systemic circulation and for the therapeutic activity. Next approach, that is pharmacosomes. In that pharmacosomes, initially that colloidal dispersion of the drugs, they have ability to bound covalently with the phospholipids. Those are available at the outer region of the vesicles, and they will exist as ultrafine vesicles, micellar systems, and hexagonal aggregates. So here you can bound that drug onto the surfaces of the phospholipids. Now they are termed as a pharmacosomes. Next approach is that transferosomes. These are termed as ultra flexible systems. They are complex vesicles, stress responsive. They will possess an aqueous core, which will be surrounded by the complex bilayer of the lipids. and they have the highest affinity to penetrate into that mammalian skin intact to the dermal, epidermal, and the subcutaneous region. So here I will give an example of to highlight on the transposome systems. Just take the example of cat. When that cat enter through the grills, those are available outside our window. The region for the entry is very limited. That time the cat will shrink their own size. They will just go enter inside to that shrinking size, and when they go enter into the house, they will occupy their own original state or that size. So that is about the best example to understand the concept of transposomes. So initially, when the transposomes are applied onto that surfaces, that time the vesicles will just occupy that cylindrical shape. They will pass to the pores, goes inside, and occupy their own original size and shape. Likewise, number of approaches are there. I just read that points, that aquasomes are there, proteosomes, colloidosomes, pingosomes. So here that initial 2 to 3 alphabets already reveal all about it. That aqua means aquasomes is nothing but the aqueous structure is there. Colloidosomes, that is colloidal systems, get encapsulated into the aqueous core. Likewise, ufosomes, enzymosomes, cryptosomes, emulsosomes are there, discosomes are there. A number of approaches are there. So I just highlight or read that particular approaches. Genosomes, genes get encapsulated. Photosomes, that photolyze. Erythrosomes, erythrocytes will be get encapsulated. Hemosomes, hemoglobin is encapsulated. Vasosomes, arachosomes, bilosomes. So these are the different types of vesicular systems. So here I added some marketed formulations of liposomes for amphotericin B with the trade name ambisome, which is specifically used for the systemic fungal infections. Likewise, doxorubicin, the myoset with the trade name, used for the treatment of breast cancer, as that permeability will be enhanced because of the doxorubicin liposomes. So I just move to that next part. Co-chelates. So this is also one of the novel approach which is available for the researcher. So we can think on it. So just zoom that slide and just check that how the drug is get encapsulated into that layers of that cylindrical layers of our liposomal structure. So when that one by one layer is opened, the drug will be come out through these chelates. Next approach is this cubosomes, that cube-like structure, and within that one cube, the drug is encapsulated. Through this each cube, the drug will be come out. So again, cyclodextrin is there out of alpha, beta, and gamma form. The beta cyclodextrin is available, through which we can having ability to encapsulate the drug inside the cavity, and ultimately the solubility of the drug is increased. But as well as these cyclodextrins are also act as a permeation enhancers to improve the drug absorption. The recent studies reveals that the another use of cyclodextrin as a permeation enhancer also. Now the dendrimers are there. These are also called as fourth generation polymers, in which the dendrimer, that is a tree-like structure, the dendron-like structure. So here the complete dendrimer is there. So within that particular cage-like structure, within that all regions, the drug will be get encapsulated, and when one by one link will be open up, the drug will be come out from the system. So likewise, at the end, whole linkages will be open, and whole the drug which is encapsulated into this whole assembly of dendrimer, the drug is available for the systemic circulation. Now, that nanoparticles all are aware about that nanoparticles as in the previous sessions, which is basically on the nanoparticles. So that nano range, that nanoscale is up to 10 to 200 nanometer. More specifically, if it will be more than microns, then it will be moved to that microspheres. So these nanoparticles are able to absorb or encapsulate the drugs, and it will protecting it against the chemical and enzymatic degradation. So ultimately the drug will be safe, and you can target the drug to the specific region of the body. So here one examples are there, that nanopores, where that pores are formed by that natural proteins or by the graphene or silicon as a silicon synthetic systems. So small tiny pores are available through which that single strand of DNA will pass easily. So ultimately that permeability will be more and more, the more and more amount of drug will be passes through that pores, and it will be enter into that cells, and it will show the therapeutic response more. Some carbon nanotubes, for that I will give one example of that. Carbon nanotubes in our old days, even in that rural area also, specifically nowadays, that ladies, they will just apply that cosmetic into that eyelids. बचपन में जो हमारी माँ हमारे आईज के ऊपर जो लगाती थी, that it is termed as that kajal. Likewise, it is the best example of carbon nanotube. So initially means it is a traditional system, but our Indian culture having all the novel approaches since, but we forget our traditional systems, and our foreign researchers just think on that particular traditional system, those are used by Indian peoples, and they will prepare and just rename as a carbon nanotube as a new name. But actually it is our old system or traditional system. As well as some quantum dots are there, bulky balls are there. Here just I highlight it, that allotrope of carbon is there, where who that 3 carbon molecules are linked to each other by single atoms, and ultimately they are again linked to each other, and at the end that bulky ball like structure will be formed. So within that bulky balls, the drug will be get encapsulated. So when that carbon bulky ball structure will be break down, the drug will be come out from that system, and it will be get released. Gold nanoshells are also there, where the drug is encapsulated and which is covered by the gold. So here, which is specifically for the treatment of anti-tumor drugs, that is doxorubicin and paclitaxel. So just mechanism of that nanoshells. So just look into that image. Cancerous cells are there. When nanoshells are administered, they will cover all the cancerous cells, and at the end, when we apply the infrared light onto that particular region, so infrared therapy is there, at that time what will happen, because of penetration of that nanoshells particles into the deeper and deeper of cancer cells, the cancer cells will be dead, and ultimately that cancers will be cured. So this mechanism will highlight how this novel approaches, that nanoshells will be useful for the cancer therapy 100% efficiently. So here some of that marketed formulations are there. Now, as I discussed previously, if the novel approaches are there with us, that time some sort of drawbacks are also there. The major concern is toxicity is there, surface interactions are there, aggregates at the periphery, and not full treatment of tumor is there, and degradation of lysosomes, degradation by the lysosomes for the nanoparticles is there. So another approach is that antigen antibody complex, antibodies drug conjugates are there, that is immunoconjugates. Microspheres are there. You can formulate that floating microspheres, sustained microspheres, mucoadhesive microspheres, hollow microspheres. Number of microspheres you can able to prepare by which you can target that specific region of the body, and as well as you can sustain the drug release by formulating the microspheres. Synthetic polymers, that is non-biodegradable and biodegradable. So here I classify different examples of biodegradable and non-biodegradable polymers. More specifically, that non-biodegradable polymers are used when you have to apply the system topically, as well as when you have to administer it orally. That time you have to operate it for the removal of that non-biodegradable polymers. Some of that natural polymers are also available. I just scroll it fastly because that time is more. So here one example is there, recyl erythrocytes. So researchers will think on that within our body's body system, that blood compartment, number of vesicles, that cells are there. So can we use the cells as a carrier? So one researcher tried to isolate that erythrocytes. These erythrocytes, just look into that image or picture. These erythrocytes, when they are added into hypotonic solution, because of that osmosis process, all you know that osmosis, the cells of erythrocytes will be get swelled, and because of swelling of cells, the pores are created, and because of pores, the drug which is available into that hypotonic solution, they get entered into that erythrocytes. Later on, these swelled erythrocytes will be pulled down. They will be added into that isotonic solution. They get washed. Some sort of drug is encapsulated inside. And here the drug loaded with erythrocytes. So now you can administer these recycled erythrocytes into the body for the targeted drug delivery. So that now we'll move to the transdermal drug delivery systems. Lot of approaches are there. So in that herpes zoster, in that treatment of herpes zoster, that transposomes gel, nanogel can be a better approach, as well as scopolamine patch is there, which is used in the treatment of motion sickness. Hydrogel transdermal patch is there, which is for the treatment of burns. Insulin patch is there for the treatment of diabetes mellitus. Again, that recent advancements are available, that is sonophoresis. So ultrasound energy is used to penetrate, to increase the permeability of the drug through the skin by using the ultrasound waves of energy or 20 kilohertz to 16 megahertz. So through that energy or ultrasonic waves, the drug will be penetrated more and more through the skin, and it will be localized into the subcutaneous region of the skin. Next approach, recent approach is laser assisted transdermal drug delivery system is there. So through that laser approach, we can rupture the cells. The drug will be come out when they get penetrated inside the skin of the stratum corneum region. Next approach is mucoadhesive systems, that is oral drug delivery system, that is mucoadhesive tablet, mouth adhesive, that particular tablet or film is there. Ocular nasals, ocucerts are there, and vaginal tablets are there. Those are mucoadhesive. So here we can just buccal tablets into that buccal region, buccal cavity. Mucoadhesive tablet is there. Nasal, that is methyl cellulose vaccine is there, which is added to the nasal mucosa. Ocular, so just ocucerts are there, which will added to that our cornea layer when they are inserted into the cul-de-sac cavity. Vaginal tablets are there, they will be added to that vagina, inner mucosa. Next approach is osmotically controlled drug delivery system is there, in which oral, parenteral, and mono, bi, and multilayer systems are available. Those are widely accepted. These systems are specifically work on that concept of osmotic pressure. And osmotic pressure is the driving force to release the drug into the controlled manner into the body. So the best example is there, that osmotic push-pull technology. Those you already studied in the bioform in that last year. Next is implantable pumps are there, through which the drug will be released. That is pulsatile release osmotic pumps. Now, what type of work is going on in the foreign countries in the recent developments of navigable system? So drug-eluting stents are there. All are you heard about that word stents? Those are used when the blockage of artery, that corona, when that hypertension is there, the heart attack is there. That time you heard about that word stent. The stent is inserted inside that particular artery. So nowadays, within that stents, the drug is encapsulated, and this drug will be eluted out from that stents as time passes. So these systems are called as drug eluting stents. See, this system, this approach is very costly, but it will be very useful for the treatment of COVID situation, because here that blood clots are there into the pulmonary tracts. So that time that the drugs or the therapeutic agents, those are responsible for the dilution of the blood, to dilute that clot, to dissolve the clots. That time the stents are inserted into that pulmonary tract region, and that time the drug will be come out from that stents, and it will be directly just affect to that clots, those are formed into that pulmonary region. So this is a drug eluting stents, novel concept on which the work is going on. Drug eluting micro stents, that micron size stents are also available. Those are implanted into that angle of iris or cornea, and this through which the drug will be come out to this polymer matrix, and it will show its therapeutic response. So here that occlusodes are there. Just look into that image. That occlusode of pilocarpine is inserted into that cul-de-sac cavity. So here that pilocarpine, which is sandwiched in between the two layers of ethylene vinyl acetate membrane, and here the drug will be come out, that pilocarpine will be come out, and this occlusode will be flowed into our eye cavity. So next one is the third generation intrauterine contraceptive devices and the norplants. So here that norplants up to the 1 year in 1992, these norplants are approved by US FDA, but year 2002, these norplants are their approval will be rejected by US FDA, because for this administration very special care and a skillful person is required, because if they are not administered carefully, that time it will lead to the another consequences. So in 2002, US FDA rejected these norplants. So this is also the novel approach. Another novel approach is microchips are there. So just our nail-like structure, small chips are there. Into that chips, just look into that image. The drug is encapsulated into that chips, and when that chips are inserted into that body, the drug will be released up to the longer period of time. So this is another novel approach. Another one is ionophores, that is our electric current at very low potential is introduced, and which is useful for the release of drug through the barriers. So again, for the delivery of insulin, so in that open loop, the administration via injections into that subcutaneous layer, and into that closed loop delivery, the closed loop pumps are available. So here the insulin pens are there, formulated by Novartis, that is Novopen. Next, insulin jets are there. Micropump also available for the delivery of insulin. Again, some sort of insulin inhalers are also there. Through nasal route, you can administer that insulin. And at the end, the recent trends in nanomedicine, that is a nano robotics. So small macro size, nano size robotics are administered. Just look into that picture. These are the robots. They directly in the treatment of sickle cell anemia. They will act onto erythrocytes. The small robots are there. They are used in the treatment of surgery, the diagnosis onto that specific cells and tissues, and they will release the drug. So for you peoples, PG peoples, I just inserted 2 slides into my presentation. How to prepare aim and objective and how to prepare a plan of work, because it is a very big task for the newly admitted PG students, that how to prepare the objectives and how to prepare the plan of work. So your basic aim is nothing but based on your formulation dissertation topic, that is to develop and evaluate the transposome based gel and formulation of antihistaminic drug. That is one example is there. So that is your final aim. To achieve that aim, you have the stepwise approach is there. So in that stepwise approach, you have to just cut down that aim into the different objectives. So how to finish your aim by different stepwise approach. So first objective is to perform the identification test on our drug as per the standards available, to perform the pre-formulation study. Next, you have to prepare or formulate and optimize the transposome system. After preparation of this transposome system, you have to optimize this system. Later on, after that, among all the vesicular systems that you prepare, you have to select the optimized one. By using optimized formulation, you can prepare the topical gel, nano gel. You have to further go for evaluation of this nano gel. And later on at the end, you have to move to the stability study as per the ICH guidelines Q1E. So this is the objectives by which at the end you will satisfy your aim. So plan of work, how to prepare the plan of work. At least first 3 months, you have to give the for the literature survey, that literature survey on system that you selected, that is nanoparticles, nano gel, sustained tablet, control tablet, whatever it may be, onto it. But among all the literature survey, around 50% of literatures must be from recent 5 years. Likewise, so you can just take a screenshot of it. So it will be helpful for you for the PG students. So these are my references. So one book is there, which is co-authored by me and our staff members, Bakliwal sir and Jain sir, which will highlight on the novel vesicular drug delivery systems, which will be helpful for you if you have to prepare the novel systems, vesicular approaches, that liposome, neosome, transposome. So all the vesicles, those are discussed today. All these vesicles are discussed in more detail in these books. So that's it. And from my side, thank you very much. So any questions?
Yes, sir. Before this question answer session, first of all, I would like to thank you very much, sir, for sharing your profound knowledge with us. Your session was very full of zeal and valuable information. As you said at the beginning of your session, that a lot of questions are there in researchers' mind about the selection of drug delivery system, particularly that the PG student has to face so many difficulties while selecting their topics, selecting their drug delivery system. And basically, particularly the last 2 slides are very helpful to PG students as well as so UG students, how to design that aim, how to design that objective, how to plan out their research studies. And you have simplified in a very well manner about novel drug delivery system, its various aspects, like its basic requirement, its principle, its strategies behind its development, then its advantages, limitations, then various applications, as well as the industrial scenario of novel drug delivery system. And also, I would like to say that you have described in depth about vesicular drug delivery system, then the recent advancements in drug devices, and as well as recent advancements in nanomedicines. So really, you have delivered the valuable information to us. Thank you, sir. Now I would like to request Ms. Namita Bhosle, ma'am, to proceed with question answer session. Yes, over to you, ma'am.
Thank you so much, Mayuresh sir. There is no any question on the chat box. Is there any questions on YouTube, sir? Bukari, sir? Okay, sir.
Participants have question related to technology.
I request to all the students, those are available. If you have any query, you can contact me at any time. I will always helpful to you. And if the PG students, in our premises, we have the different research facilities. So we will be helpful for you for GCM, SMS, LCMS, for preparation of nanoparticles. As we have that Panda high pressure homogenizer with us, high speed homogenizer with us. So in collaboration, even with your staff, those are engaged with our PhD program and they wanted to develop that nanoparticles. So I will be just from our college, we will just take a permission and we will be proceed for our collaborative work. Thank you. Thank you very much.
Thank you so much, sir. Now, dear participants, this is the lunch time now. We will rejoin the session exactly at 2:30 p.m. So after lunch, we will have our remaining technical sessions.
1 second, madam. I am very much thankful to principal of the institute, Dr. Jagtap sir, the coordinator of this symposium, national level, Dr. Dattatreya Shinkar, for inviting me into that national symposium and providing me the opportunity to discuss with the new generation researchers on that nano concepts. So thank you very much, and all the organizing committee, more specifically that Mayuresh sir, Rukari sir, and Namita madam. Thank you very much.
Yes, thank you, sir.
Thank you so much, sir.
Thank
So
you,
it's
sir.
a request to all the dear participants. Please rejoin the session exactly at 2:30 p.m. Enjoy the meal. Thank you.